pleasure to welcome you on the second day of our forum. Today we will have the second symposium on precision medicine. This symposium has been organized and will be chaired and moderated by Dr. Colin Brennan and Dr. Ali Tnazlı. I'm going to introduce them briefly. And Colin Brennan, he's currently Vice President of Technical Activities of IEEE. He's also the founder of Hi-Fi Bio, and I call him the builder, actually. He built several companies, very successful companies. And also, I'm pleased to mention that he's chair of Industry Advisory Board of University of Houston Biomedical Engineering Department. Ali Tnazlı, Dr. Tnazlı, he is the currently Chief Commercial Officer of the Flux Energy. He was with HP, with the global division, global head of the division. And before also, he was the CEO of the bioscience of the Sony. He is going to expand his company. We just learned this morning that they are going to have also the satellite, the campus in Germany. So Colin and Ali, I appreciate all your efforts on behalf of IEEE, and also I appreciate all our fantastic, great speakers this morning. Colin,
well, thanks. Oh, thanks very much, Ed. That's a wonderful introduction, and we thank you for your leadership of the society. Those are very kind words. So myself and Ali are co-organizers of this symposium, Precision Medicine and Data Science and Engineering Healthcare. We welcome all who have joined us through the webinar. Good morning and good afternoon, and perhaps even good evening. So just to frame today's discussion, that I thought I'd give you a few remarks and thoughts on precision medicine and related to data science. So as you probably already know, that precision medicine is an emerging framework for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person, that enabling doctors and researchers to predict more accurately which treatment and prevention strategies for a particular disease will work in which groups of people, with the goal of delivering the right drug to the right person at the right dose, at the right time, and by the right route. It is in contrast to a one-size-fits-all approach, which in disease treatment and prevention strategies are developed for the average person, with less consideration for the differences between individuals. So we'll learn today from our illustrious and expert panel of speakers about how data science intersects, impacts, and enables the fast-changing field of precision medicine by applying or inventing the tools, techniques, and theoretical underpinnings needed to enable application of machine learning and artificial intelligence to large, diverse, complex, longitudinal, and distributed data sets, typical of precision medicine, to extract useful information and knowledge so as to transform healthcare and delivery and fundamentally our healthcare industry on a global basis. So the format for today is we've asked our speakers to plan to speak for roughly 20-25 minutes, followed by a few minutes of Q&A from the audience, moderated by myself or Dr. Tnazlı. Our first speaker is Professor Raimond Winslow from Johns Hopkins University. And I ask my colleague, Dr. Tnazlı, to introduce Professor Winslow.
Hello, good morning, everyone. Also, thanks to the team here and to Metin for his leadership. And it's an honor to co-chair this session with Colin. So I'd like to introduce our first speaker for the day. It's Raimond Winslow from Johns Hopkins University. And he'll be basically speaking about predictive analytics in critical care medicine. And as we are experiencing during this pandemic times, critical care is increasingly important and really just highly aware, I think, on a society level. So I'm looking very much forward to learn more about his work and the latest from his thoughts on predictive analytics. Raimond.
Thank you. So thanks for the introduction, Ali, and Metin and Colin and Ali for the invitation. I'm gonna, okay. How do I go about sharing, host disabled participant screen sharing?
You go basically.
You should be able to do it
the share screen. There's a it's like, yeah, it's like
okay.
there's a green button.
Yeah, I've got it now.
Share screen with an arrow. Yep. Here you go.
Yeah,
Perfect.
it's working. Thank you. Thank you. All right. So as Ali said, I'm going to be talking about some data science applications in critical care medicine. And let's start off with the why. So every day when an intensivist, a critical care physician, sees their patient, like other physicians do, they ask three fundamental questions. First of all, what's the state of my patient? Second is they're looking at information about their patient and they're wondering and hoping to understand if the patient is getting better, if they're getting worse, or they're going to stay the same. And then the third thing they need to answer is, how should I treat my patient given the condition that they're in and given what I think is happening to them? And I think each of these three questions are opportunities for data scientists and engineers to work on and contribute. And so here's a picture of an infant in the pediatric ICU. It's a very gripping picture because it shows how, of course, anyone in the ICU, these are among the sickest patients in the hospital, but the point of the picture is how heavily patients in ICUs are instrumented and how many different kinds of sensors are used to monitor physiologic function. And so the ICU for the intensivist and other caregivers is a really data intensive environment with many complex signals being monitored in real time and displayed for them to examine. And intensivists are faced with an enormous challenge to interpret these data. There's so much data. They struggle with how to take it all in and interpret it and turn it into actions that can help their patients. And so intensivists and engineers like myself think this is an opportunity for data science approaches and engineering methodologies to contribute in critical care medicine. And the opportunity specifically is to address these three questions that intensivists and other physicians ask every day with every patient. So I believe that engineering methods can be applied to help quantify what is patient state, and I will give specific examples of how we do that, very simple ways of doing that, how one can predict the evolution of patient state to make the kinds of forward predictions that are needed to really quantify and answer the questions, is my patient getting better, worse, or staying the same? And engineering methods can also guide therapeutic treatment. So I'll talk in depth about the first two bullet items. I'm only going to touch on the guiding of therapeutic treatment in a kind of indirect way, but data science methods can very clearly, well, I will say clearly have the potential to help here. Everything we do in the critical care unit needs to be tested, of course. It needs to be proven in clinical trials with real patients to work. And so I want to talk about three case studies, and one I'm going to talk about in a fair amount of depth, and that is predicting in adult patients who have sepsis, which of them will progress to septic shock. So the given is these patients have sepsis. We want to watch and monitor them in an intelligent way to predict those going into shock. Just to remind you, sepsis and septic shock, its most extreme form, are very severe systemic infections that lead to reduced blood pressure, reduced organ perfusion. They can lead to organ failure, and they can lead to death. Estimates of mortality in the US range from 20 to 50%. So without a doubt, it's a very significant and highly lethal disorder. And the estimates are that it accounts for 30 to 50% of all hospital deaths. It's a very expensive disease to treat because of the numbers of patients involved. And it's also a leading cause of hospital readmission. So by any metric, sepsis and septic shock is a serious condition and the one that needs attending to. So we know that early treatment helps. An interesting observation is that mortality in septic shock increases about 8% for every hour of delayed treatment. And when we saw that fact, we realized it was telling us something about the natural time course of evolution of the disease. It clearly evolves rapidly. If mortality, once you're in shock, increases 8% every hour, it's a very rapidly evolving disease. And so the data that one harnesses to predict ahead of time the occurrence of this event must be data that can be acquired rapidly to match that time course. We know that early goal-directed therapy, which typically involves administration of antibiotics, fluids, first of all, fluids in an effort to bring patient blood pressure up, often vasopressors to elevate blood pressure and increase organ perfusion, and antibiotics to fight infection are in early goal-directed therapy. The goal is to administer these therapies as quickly as possible on suspicion of sepsis and detection of septic shock. And so our challenge in our work is to provide early warnings in sepsis patients of those who will go into shock. And the reason for doing this is we want to open up a window of time in which physicians can intervene with early goal-directed therapies to try and prevent shock from occurring. And here's how we think about the task at hand. This is how we formulate it. It's our key hypothesis. It's incredibly simple. In hindsight, it's incredibly simple. This axis is time. This axis is what we call a risk score. A risk score is a probability, so it ranges from 0 to 1. And in this case study, this risk score is the probability that a patient will enter septic shock at some future time. So we're sitting at time t. This is the probability that at some future time point, the patient will transition to septic shock given they're in sepsis. So we hypothesize this risk score, which I've just sketched here. This is not a real risk score. And this illustration, during this time, the patient is clinically diagnosed as being in sepsis. At time T2, they meet the criteria for septic shock. And so they're in the state of septic shock during this time. We hypothesize that there exists a threshold on this risk score such that if the risk score exceeds the threshold, we'll make a prediction that this patient with sepsis goes into septic shock. And we're hoping that there is a threshold that enables this prediction to be made reliably. And if we can do it reliably, then the time interval between the onset of septic shock and when we make this prediction, hopefully we want to prevent septic shock by making this prediction, but in retrospective data, this window we call the early warning time, and that is the advanced warning that in principle we could give a physician to attend to this patient and perhaps intervene if their judgment is such that they choose to intervene with antibiotics, maybe change the antibiotic course, give fluids, give vasopressors. So this is the idea. And we call this state here, in this particular application, the pre-shock state. And note that the patient is still in sepsis in this pre-shock state, but if they enter it, then it's extremely likely that they're going to transition into septic shock at some time point in the future. So here's the clinical definition of sepsis and septic shock that we used in this study. It's known as the Sepsis 3 consensus definition. There's an international body of physicians who meet regularly to discuss and review the clinical definition of sepsis and septic shock. They've met 3 times and published papers on their consensus definitions. Sometimes they make revisions, so the Sepsis 2 definition differs from the Sepsis 3 definition in actually very important ways. But this is the one that's in play now. It's the one we adopted. We say that a patient is in the state of sepsis if an infection is suspected on the basis of blood tests and the admission and the treatment with antibiotics, for example, and 2 or more of the following conditions apply. These are conditions on respiratory rate, systolic blood pressure. Obviously, if blood pressure is dropping, that's an aspect of sepsis, and an assessment of neural function known as the Glasgow Coma score. If that score is below a particular threshold, then the criteria are met. So 2 or more of these criteria and the sequential organ failure assessment score must be greater than a marginal value of 2. And this means that there's some indication that organ failure may be beginning to develop, or a patient is at risk for organ failure. In septic shock, a patient must be in sepsis, and vasopressors must be needed to maintain mean arterial pressure. In serum lactate is elevated above a threshold value. This differentiates shock from sepsis. Elevated serum lactate is an indicator of low oxygen perfusion, reduced perfusion of into the logistic function, and we estimate the probability that this patient at time t with sepsis will transition at some future time to shock. And as a principled way of getting the best betas, it's maximum likelihood estimation, maximizing the log likelihood function of the data where the class labels are assumed to be 0, 1 Bernoulli random variables for sepsis or shock. All right, so to say a little bit more about the features, we use features that evolve at a minute time scale: heart rate, systolic blood pressure, arterial oxygen level, and features from the electronic health record, which are entered irregularly and typically at much less frequent intervals than are these numeric data. And so every time a new feature is available, we update the risk. And we are in quest of a threshold on that risk that that results in a decision that this patient with sepsis will go into shock. And we want to know if a threshold exists that allows us to do that reliably. So here is a receiver operating curve for that binary detection, binary classification tasks using 3 different models. This green line here is obtained using a recurrent network. I showed you the logistic regression function. This is in black. You see the receiver operating characteristic curve for that way of building the risk function. And we took as the best theta the point on the receiver operating characteristic curve that yields 0 false positive, that is the point that is closest to the point of 0 false positive rate and 100% true positive rate, the point of perfection. So roughly a point like this. Every point on this receiver operating curve is determined by a choice of threshold and computing the corresponding specificity and sensitivity for that choice of threshold. So choice of this point on the ROC curve determines our threshold. And using that threshold, we can detect and predict impending septic shock with 88% sensitivity, 84% specificity, area under the curve, an overall positive predictive value of about 50%, but I'm going to say more about this in just a moment. And the median early warning time in this task is 7 hours. And so that's sufficient time for a physician to intervene and do something about this. Now, to positive predictive value, we found something interesting. So there's several interesting things in this plot. So this is a real patient risk score. Note one thing about this real risk score is that the rate at which it crosses threshold is different from what I showed you in the hypothetical example. The threshold crossing occurs very rapidly in this patient. We'll return to this idea in a bit. I want to define the distance from the threshold to the value of the risk immediately after crossing threshold. We'll call that D. What we did was to break the range of risk values from the threshold to the value 1 into 10 different equally spaced bins, looked in our training set at the magnitude of this distance D for that patient and assigned them to different bins. And so in some bins, the initial risk value is very high, and others it's very low. We then went into those bins and looked at the reliability with which we could make the prediction that this patient is going to septic shock. The higher D is, the larger D was, the more reliable was that prediction, the higher the positive predictive value. And there was a linear relationship between the percentile of D that you were in and positive predictive values. So if this jump is really high, when you cross threshold, you can be making these calls with up to 90% reliability. The fraction of actually true calls that you make is very, very high. Whereas if this jump is small, the reliability is less, is smaller. So not only can we make a call, but we can make what we call a patient-specific positive predictive value and provide that to the physician. We can say, we estimate we're 90% confident of the call, or 20, or maybe 10% confident of the call. And that's certainly going to affect, in principle, the way physicians respond. Now, let me go back. So what we did here was to consider all of the temporal profiles of risk for patients whose risk profile crossed threshold, some of whom would go into shock, others didn't go into shock. But we took all of those that exhibited a threshold crossing, lined them up at time 0, which is the time of threshold crossing, and applied a technique called spectral clustering. Spectral clustering is a way of looking at this ensemble of time waveforms, the risks are time waveforms, and asking the question, how many naturally occurring groupings are there? How many classes, broad classes of time waveforms are there in this ensemble? In this particular analysis, the answer was there are roughly 4 classes. And what you see here in the darker lines are the average values of the risk waveforms within those 4 classes of risk. So there are several important things here. The first is look at the average time waveforms and its the average risk scores and their confidence intervals before threshold crossing. All the patients look the same. You can't tell them apart. The second important thing is what I showed you earlier. The risk score increases dramatically at the time of threshold crossing. Something dramatic is happening in these patients. The variables that we're measuring from them are very suddenly driving the risk very high over a brief period of time of something like 30 to 60 minutes. And it's following the threshold crossing that these patients stratify into different groups according to their risk waveforms. There's a high risk group. We call it high risk because 77% of them go into shock. 43% of them unfortunately die. And then there's some other treatment variables that I'll talk about more momentarily. But look at the percentage of the patients within these 4 groupings that go into shock. It's highest for this high risk group. That's why we call it the high risk group, but it decreases monotonically as we sweep across the other groups. Look at mortality, that decreases monotonically. This measure here is, we ask the question, what fraction of patients in each of these groups are considered to be adequately fluid resuscitated by the time of threshold crossing? It's lowest in the high risk group. Arguably, they're getting less fluids than our other patients. and it increases monotonically to the low-risk group. We asked the same question of vasopressors. What fraction of these patients have been given vasopressors before the time of threshold crossing? It's smallest in the high-risk group, and it's highest in the low-risk group. So remember, these are treatments for sepsis. And it makes sense to me that for whatever reason, the high-risk group has the lowest fraction of patients who've been adequately fluid resuscitated and adequately have received the smallest fraction have received vasopressors by the time of threshold crossing. And conversely, it makes sense that we see the kind of opposite relationship for the low-risk group. But I consider the most important information in this spectral clustering analysis to be, even in the ensemble of risk forms, the threshold crossing is very, very rapid. It says to me that something is happening biologically in these patients. They're kind of collapsing over a brief period of time, and their risk is shooting up. And then they're very likely to transition to shock. The final thing I will say about this is maybe whatever is happening here, maybe this is fundamentally septic shock. It's not the event that occurs out here, maybe 10 hours in the high-risk group on an average. It's not the clinical definition that I gave you earlier. That's what I'm struggling to say here. Maybe this event that we have defined computationally, maybe this really marks the fundamental event that signals septic shock. So quickly, I'm going to show you just a slide each on other case studies. Here we're predicting pediatric sepsis patients who will progress to septic shock. We did it in exactly the same way that I talked about. We can find a threshold that gives good performance in this classification task. The median early warning time is about 9 hours. We can do this classification very reliably. The spectral analysis of the risk waveforms shows exactly the same thing. No distinction between waveforms before threshold crossing. The threshold crossing event is very abrupt. Patients stratify following the threshold crossing event. Here we are looking at this in children in the PICU, and we're trying to predict those who will develop multiple organ dysfunction syndrome, a highly lethal disorder and syndrome in the PICU. So here's the receiver operating characteristic curves. The performance in doing this task, it's high. That is to say, we can find a threshold. The early warning time is 37 hours here. We can reliably make this prediction ahead of time in 37 hours. The spectral clustering analyses show the same kind of results. The Goldstein and the Proul criteria are just different clinical criteria for defining multiple organ dysfunction syndrome. There's not a consensus definition of this syndrome. Multiple definitions are used. Here they are. We looked at building these risk functions using various methods, but you see they all yield about the same results. There's an abrupt change in risk at the time of threshold crossing, and then we stratify into maybe 2 or 3 groups. Importantly, there's a very high-risk group and a low-risk group. What's remarkable is our positive predictive value for the high-risk group is 97%. It's extremely high. If you're in this group, you essentially will develop multiple organ dysfunction syndrome. And with a median early warning time of 37 hours, maybe that's providing enough time to intervene and for physicians to help better treat these patients. So with that, I want to conclude by stating my favorite phrase, which is medicine is becoming an engineering discipline. This is how I see it. And what I mean by that is that physicians will always be at the tip of the spear making healthcare decisions to treat their patient. But increasingly, these decisions are going to be informed by data science methods, in general by engineering methods, by models that are running in the background, or maybe not even in the background, are running to provide information to physicians that will guide their decisions, just like a lab test guides their decisions. And a corollary of this is that I believe physicians' training needs to adapt to this inevitable transition, that they need to know core elements of data science, because it's going to be playing an increasingly important role in medicine over the coming decade. I see this transition underway in critical care medicine. Intensivists see the value here of these technologies and approaches and methodologies. So what I've showed you here, remember, are just retrospective analyses. We are not running these algorithms clinically. We are not informing physicians about what's going on so that they can adjust their treatments. We need to do that. We need to pursue the hard task of bringing algorithms into the clinic and testing them to see if they work. But in doing this, I think we have some major opportunities to contribute to healthcare and to help save lives. That's all. Thank you very much.
Thank you, Dr. Winslow. I really enjoyed your talk. Thank you very much for bringing this important topic to attention. And in the meantime, as we may get some questions from the audience, I'd like to start actually with the question, when you look at the intensivist workplace, how do you see from a technical perspective new opportunities originating from point of care and IoT? Because I think IoT will be one of the key enablers for better, more comprehensive longitudinal measurement in order to just keep a better line of sight of the individual patient. But it's like, where do you see this in terms of technology solutions coming? And second, how close is it to the real world clinical setting on an average community level? Maybe a bit too complex, my question, but again, perhaps this also helps the audience to think of, let's say, questions in this field, because I think this can be quite exciting.
Great questions. Thank you. Yes. So remember that 3 of the variables I showed you were what I called vital signs that were made available on a minute-by-minute basis. One remarkable thing, this is a true statement, is that in most hospitals in this country, those minute-by-minute vital signs data are not grabbed and stored and archived. They come and go. And every now and then they are inserted into the electronic health record. And in virtually every hospital in this country, with few exceptions, the waveforms that are displayed on the monitor are ephemeral data. They're there and gone and lost forever. So technology solution 1 needs to be capture these data. I think they're incredibly important for learning systems and for understanding how to diagnose and treat patients better. The low-hanging fruit is clearly the minute-by-minute vital signs data, which are in principle available in every hospital through HL7 messages, a standard of data communication in hospitals. What is lacking are technologies for grabbing those data continuously in an automated way from every single patient in the hospital so that they are available for use in algorithms like the one I've described. So that's one part. We need that technology. The second thing is that we need ways of test. So I see a staged approach to deploying in the clinic. What we're working on now is something I will call delayed real-time analysis. So what I mean by this is that at Hopkins, every 24 hours, the contents of the EPIC database are rolled into EPIC Clarity, a research database, and into another system called the Precision Medicine Analytic Platform. So with 24 hours delay, all clinical data is available for research purposes in PMAP. We've built a software platform called PhysioCloud that collects the minute-by-minute vital signs data in an automated, scalable way for every patient at Johns Hopkins Hospital. It's now the clinical information system for these kinds of data at Johns Hopkins. That is continuously available along with the delayed EHR data in PMAP. So what we're working on now is implementing these algorithms in real time, but with this 24-hour delay. So patients may still be in the hospital and we're tracking them and we're making predictions about their state and how it will evolve over time, but we're doing it in a prospective way without feeding back information yet to physicians. And the reason for doing this is that it enables us to understand how to build platforms that can do this effectively in real time, and how to address questions of how do we go about feeding this back to physicians. The final step, of course, is to bring this into real time and interface not to Epic Clarity or PMAP, but to Epic, and be pulling the variables that we need out of the EHR in real time, along with the physiologic data that we already have in real time, to be running these risk scores and to be notifying caregivers when transition events are predicted by these algorithms, and to hand those physicians and caregivers a reliability of that prediction so that they can weigh that reliability and decide, well, if the call is this patient is going into septic shock, and with 90% confidence, I better do something now. But if the call is maybe the confidence is 10% that this patient will go into septic shock, I would certainly want to attend to that patient because they're at risk, but maybe the treatment decisions will be moderated and modulated by the physician armed with that reliability. So that's how I see this evolving.
Thank you very much. I think we should let's definitely discuss further. That's a very exciting field. And then we got a question from the auditorium. There's a question from Kanita. She's asking how you see the future curriculum of medicine or pharmacy. Do you think it is mandatory now for medical students or pharmacy students to study more on AI or data sciences?
That's a wonderful question. I've thought a lot about it. Let me answer by saying that at Hopkins in the biomedical engineering program, our master's degree program has focus areas, and we've defined, this is illustrating an answer to your question. We've defined an area we call AI in medicine, and we have another area called biomedical data science, but AI in medicine is specifically for medical students doing a gap year, residents, clinical faculty, and we have all three of those categories enrolled. This is our first year off this. We have all three types of people enrolled in this program. And so what's the goal? What's the purpose of the program? It is not necessarily to create expert leading edge data scientists, although of course we want to do that if possible. I think our real goal is guided by the fact that more and more papers on machine learning and artificial intelligence in medicine are going to be appearing in New England Journal of Medicine and JAMA. They're already there, and it will only grow. And physicians need to understand how to read these papers and interpret and be critical of the results, and know how to answer the question, what does this really mean for the medicine that I'm doing? Just like they look at the experimental or clinical research literature and critically evaluate it and assess for themselves how they want to use that information in their practice. We want to equip physicians to be able to do this. With data science methods. And so we offer this master's program. It's very interesting. A trend in medicine is that many medical programs are becoming de facto 5-year programs. Nationally, 60 to 70% of all medical students take a gap year now to do something else. And we saw that as an opportunity to capture these students and train something and offer them something new in data science. So I think there are clear ways in which data science will emerge in the medical curriculum, even within the traditional 4-year or sometimes 3-year medical curriculum itself. So that's an incredibly great question, thanks. And there's a lot to say about it. I could say much more, but I hope those examples help answer that question for you.
Thank you, Dr. Winslow, for answering this. Yes?
I have a quick question for Dr. Winslow. So at what point is, this is fabulous. It's really, really very interesting. I was kind of curious, at what point does this predictive, these predictions become actionable? Because you had mentioned it to a certain extent. So, are internists already acting on this information, or is it's still too early for them to be able to use this information and to be able to address the issues associated with each of the different at-risk groups?
In our particular case, it is too early. We're not providing any clinical feedback to physicians. There are institutions where analogous types of algorithms are becoming deployed and are informing physician decisions. And so I think the unique thing about critical medicine is how welcoming critical care physicians are to this approach. They know that they can benefit from this kind of assistance. They love to work with engineers on these kinds of problems. And I know that they will attend to and be receptive to the calls we make. And of course, they'll be rigorous and critical of the value of the calls we make, the warnings that we can provide. But I think we're, in our personal case of our personal research, we're probably about a year away, I think, from being able to go real time and inform physicians about events that are happening. And I think increasingly, this is going to be a mainstream aspect of critical care medicine over the coming decade.
Okay, thank you very much. I appreciate it. So this was great. So Colin, do you want to proceed then with the next speaker?
Yeah, thank you very much.
Okay, thank you again.
Wonderful.
Thank you. Yes.
Raimond, thank you so much, my friend. Thank you.
Thank you.
So it's my pleasure to introduce the next speaker, Craig Cummings from Genentech. Craig is the Senior Director and Head of Data Science with Oncology Biomarker Development at the company. His group is responsible for development and execution of global data analytic strategies, including bioinformatics, real-world clinical genomics, liquid biopsies, and digital pathology to accelerate translational research and drive early and late-stage oncology developmental pipelines. Prior to joining Genentech, Craig worked at Sequentia, where he developed tools for NGS-based minimum residual disease detection in lymphoid cancers and T-cell repertoire analysis, and at Applied Biosystems Life Technologies, where he was responsible for pathogen detection assay design and NGS-based microbial genomics applications. As a postdoctoral fellow and research associate in the Stanford University Department of Microbiology and Immunology, Craig investigated Bordetella population genetics and gene expression using microarray-based approaches. He holds a PhD in genetics from the Stanford University School of Medicine, where he was HHMI predoctoral fellow, and a BA in biology from the University of Virginia. So it's my pleasure to invite him here today. And the title of his talk is Deriving Personalized Oncology Through Advanced Analysis of Genomic, Cellular, and Image-Based Biomarker Data from Large Clinical Trial Cohorts. Craig, the floor is yours. Oh, you have to unmute yourself too.
All right,
perfect.
Can you see my slides too, Colin?
Yes, perfect.
Perfect. Yep. All right, I've mastered the technology. Thank you, Colin, and thanks to all the organizers of this symposium for the invitation to speak here. I'm really excited to have the opportunity to present. So, I work at Genentech, and I'm a biologist. I am not an engineer. I'm going to present examples from the literature, including some works that my colleagues have contributed to, and also from different academic groups, just to really give an overview of the kinds of things we can do in this space. But before we dive into biomarker data analysis, I'd like to start with a very brief introduction to cancer. It's really a very diverse collection of diseases with widely different prevalence, outcome, and treatments. But all cancers have some basic mechanisms in common. So in normal development, your cells know when to grow and when to stop growing. So in utero, going from a single fertilized egg to a fetus, obviously this is very rapid growth. This happens all over the course of 9 months, and a whole body plan is laid out. So there's a lot of cell division, a lot of cell growth. Now, once that child is born, they're still growing, not nearly as rapidly, but it's slower growth in selected organs. And then once you get to be an adult, you basically grow when you need to grow, for wound healing and such. So it's a highly regulated process, and again, this is normal development. This is how things are supposed to work. Now, cancer happens when mutations cause your own cells to divide and grow out of control. So what happens is a cancer-causing mutation arises in a single cell, and in some cases, that will result in either an abnormal protein or loss of a protein that's important to regulating cell growth. That leads to rapid growth and cell division. And then ultimately, as that tumor grows, it disrupts the tissue architecture and goes on to become what we would call a tumor. So this is a highly dysregulated process, unlike our normal development. So, the key question that I want to try to ask today is, why don't oncology drugs work in every cancer patient? So reason number one is that cancer is really a very heterogeneous disease. So lung cancer can be separated into multiple histologies, each with multiple molecular subtypes that are targeted by different drugs. So over time, lung cancer classification has become increasingly granular. So non-small cell lung cancer, which is, so let me back up. Lung cancer can be broken into two different histologic categories. One is called non-small cell lung carcinoma, and the other is small cell lung cancer, or small cell lung carcinoma. So if we just take this non-small cell lung carcinoma piece of the pie and break it down further, you can see that there are at least three major histologies here: adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. And then there are others that aren't specified. Now, we can break this down even further into genetically distinct subtypes. So if we just take the adenocarcinoma, for example, we can see now, based on a lung adenocarcinoma study from the TCGA, that there are many different molecular classes of adenocarcinoma. So some are driven by a gene called KRAS, some are driven by mutations in EGFR or BRAF, and then you can see a whole bunch of very tiny slices, all different molecular drivers here. And you can see the prevalence of these is quite rare. So these molecular subtypes could all require a different treatment paradigm. So, what you can see is that, what you might think of as a single cancer type, lung cancer, is really many different tumor types defined histologically and molecularly. And this really presents a challenge for how you treat them. So now I'm just going to show a quick example on further partitioning of small cell lung carcinoma. And this is from a study that some of my group participated in with the MD Anderson Cancer Center, and you'll see I have the links to the articles on each of these from which the figures are derived. But they applied non-negative matrix factorization, which is an unsupervised clustering algorithm, to identify gene expression-based small cell lung cancer subtypes. And what you can see here is, if you take this gene expression data here, which is colored by expression, with blue being low and red being high, you can find these distinct clusters up here, 1, 2, 3, and 4, that each have a different gene expression pattern. And if you go in and look at this data, you can deduce that each of these different groups, or at least 3 of them, are being driven by 3 separate transcriptional factors, these 3 right here. And if you go in and look at the expression of those transcription factors, you can confirm through an imaging-based technique where you're staining for the expression of those proteins, that those 3 different clusters, or those 4 different clusters, 3 of them express those 3. Oops, I'm sorry, I need to go backwards. Those 3 the transcription factors. There's also a 4th type that comes out here that actually doesn't express any of them. So, this is one that we know less about, and that, we know that it's immune inflamed, but, this is an area where this is discovered a new group that we didn't really understand well before, and we want to explore that more. So these actually have clinical ramifications. So this study, I'll say right off the bat, was not powered to address this hypothesis. But what you can see is that in one of our studies where we combine chemotherapy with a drug called Tecentriq, which is an immunotherapy drug, and compare that to chemotherapy plus placebo, we find that this immune enriched group, which we call SCLCI, actually performs better. So this is a survival curve. The further to the right they are, the better they survive. So you can see between the Tecentriq treated and the placebo treated, we do see a difference here. And it's not statistically significant. Again, this is not powered to answer this question, but it certainly trends in the direction that we would expect since this Tecentriq drug targets the immune system. So here's yet another example in a different cancer type called renal cell carcinoma, where again, we've applied NMF, non-negative matrix factorization, to identify gene expression clusters. There are 7 of them that we found in this disease. And when we look at the biological pathways that are expressed in each of those 7 clusters across the bottom here, you can see that they're quite diverse in the different biological pathways that they are expressing. So, these appear to be biologically different. If we look down here at the bottom at the mutation profiles in each of these clusters, each of these rows is a gene, and when there's a color, each column is a patient, and when it's colored black or blue or another color, that means there's a mutation there. So what you can see is that each of these clusters, differs in the prevalence of these different mutations. So, for example, VHL is highly prevalent in this disease, but here's a cluster 5 that has many fewer VHL mutations than you would expect. You also see that CDKN2A and 2B are regulators of the cell cycle, and when those are deleted, which is the blue color here, you can see that those are those result in high cell proliferation. So it makes sense that they are enriched in this proliferative cluster here. But what this means is that you would potentially treat these different subtypes with different drugs. There are inhibitors, for example, against the cell cycle components that are regulated by CDKN2A and 2B. So you could potentially hypothesize that these patients would respond better to those drugs. And again, if you look at the response of these different subgroups to different combinations of drugs, so atezolizumab plus bevacizumab versus another drug called sunitinib, you do see differences in how these different subtypes respond. So, for example, this one here in the kind of yellowish gold color survives very well in this treatment regimen, whereas it's kind of middle of the pack when treated with this drug. So the second reason that not all cancer patients respond the same is that tumors are genetically heterogeneous and continuously evolving. So there are more than 30 trillion cells in the human body, and they divide about 2 trillion of them divide every day. So, this is just a schematic of what replication from a single cell looks like. So in organ development, this is normal development. Cells are dividing, and DNA replication is imperfect, but the vast majority of the times it won't go on to cause a mutation that will go on to cause cancer. Most of those mutations are silent. The cell just doesn't care. But over time, sometimes cells will acquire mutations that allow them to grow more quickly and gives them a growth advantage. So that leads to these clonal sweeps and having precursor lesions arise. And then as additional driver gene mutations are acquired over time, these can go on to become fully transformed and become a tumor. Now, once a tumor is actually established, these cells tend to get increasingly unstable in their genomes, which leads to more mutations and may lead to different subclones arising in the tumor, some of which go on to spread to other parts of the body and through the process of metastasis. And I'll point out here that this heterogeneity of the tumors also provides a deep reservoir of resistance mutations, which I'll talk about shortly. So there's been a real explosion in the ability to assess tumor evolution over time. So in 2012, this is a seminal paper from Charlie Swanton's lab at the Cancer Research UK. And he took multiple tumors out of a kidney cancer patient, both the primary tumor as well as metastases, which are labeled with M throughout here, and sequenced all of them, or a number of metastases, and then different parts of the primary tumor indicated here. And what you can see if you look at the distribution of mutations throughout the different samples is that they are genetically distinct. They have mutations in different genes, which are across the top here. And what that means is that you can use this data just like you would use any other mutation data to build phylogenies and understand the evolutionary history of that cancer. So, for example, this is one patient here. You can see, in normal tissue, it acquired a VHL mutation, which is common to all of the samples. And then you can watch it branch. In this case, each of the two branches picked up different set D2 mutations that likely have the same consequence, but go off to form different subclones, which then pick up additional mutations as they go. So, this is really the first paper to show that this can be done systematically. Since then, there have been many studies, but one study to or one method to assess heterogeneity that's been increasing in popularity is single-cell genomics. So this is a study from the Broad Institute where they explored genomics copy number in single cells and gene expression from 5 glioblastoma tumors. So on the left here is the copy number profile. Red means that additional copies of that locus have been gained, and blue means that they've been deleted. And you can see in each of these tumors, which are indicated by these different horizontal blocks, there are some common mutations that you see in all of the tumors. For example, this chromosomal deletion is found in all of them, but you can see that they're all genetically distinct in terms of their copy number profile. And even within a single patient, we can see different clones have different copy number profiles. If we look over here at receptor gene expression, so for all these, a selection of different receptors, each row represents an individual cell, and we can look at expression of those genes. And you can see each of these cells within the tumor is actually expressing a different set of receptors. So this, genetic heterogeneity also leads to gene expression heterogeneity. So the second study here from MD Anderson shows genomic heterogeneity in a single AML, acute myelogenous leukemia sample, from which the evolutionary trajectory can be inferred. So again, similar to the multi-region sequencing study that I showed you, we can go and identify different subclones that have different mutations and then build phylogenies like this that show the evolutionary history of the tumor. So this is an interesting study from USC using single-cell sequencing to look at a cancer cell line. We often think of cell lines as being homogeneous, but even within a cell line, you can see differences in copy number throughout this population, and you can find 4 clusters and actually identify events that happen within this, this group of cells in a plate. So heterogeneity is really intrinsic to cancer. So the consequence of heterogeneity in evolution in cancer is often treatment resistance. So this is an example of a patient who had BRAF V600E mutant metastatic melanoma, and they were treated with Zelboraf. So each of these lesions is a melanoma metastasis in the skin. And this patient was treated with Zelboraf, and at 15 weeks had this really dramatic response. This is, what we, this is everything we would hope for in a targeted agent. But unfortunately, at 23 weeks, almost every one of these lesions has relapsed. So this is really a common problem in personalized targeted therapy, is that, you know, even if we have the right drug, the tumor finds a way to outrun it. So I just want to show that we can track subclonal evolution, not just in the tumor, but also in circulating DNA. This was a collaboration with Trevor Graham's lab at the Bart's Cancer Research Center, using some data generated in our group at Genentech. And what we can see here is, looking at longitudinal data for 3 different patients, that over time, we can see quantitative changes in the prevalence of different subclones defined by their copy number profiles. And they're summarized in these bar graphs at the right. So you can see this is after treatment with a drug targeting the PI3 kinase pathway. You know, at baseline, there's this baseline profile, and then you can see after treatment that you know, we see new clones arising, and we see increase in subclones that may have been there to begin with. And then another example, this isn't using copy number profiles, but rather targeted mutation analysis, is tracking emergence of resistance in patients from one of our trials who were treated with another drug targeting the PIK3CA gene. And what you can see is that over time, these patients, they acquire new mutations in the PI3 kinase and the ESR1 genes, which I'm showing here. And in many cases, they're acquiring multiple different mutations in their bloodstream. And we can track this over time. So if you look at the clonal heterogeneity of different mutations in individual patients, so each graph is a patient, you can look at the dynamics of these emerging resistance mutations. And this is potentially important clinically. If you have a patient who you can screen longitudinally and see resistance mutations coming up in their blood, maybe you would switch their treatment paradigm. So the third reason that tumors, that not all patients respond to cancer therapies, is that tumors are embedded among normal cells and manipulate their function. So tumors aren't just a ball of cancer cells. They organize so that they have a blood supply for oxygen and nutrients. They recruit fibroblasts and other cells to create an optimal environment for their growth. Immune cells often come into tumors and try to kill tumor cells. And in fact, they probably do this all the time in our bodies. But sometimes tumors evolve ways to hide from the immune system. And this is an example of one way that we know is very important. They express a protein called PD-L1, which is basically a stop sign that tells the T cells that are coming in to try to kill that tumor that, you know, don't bother, we're safe. So they kind of cloak themselves. So based on that, you know, there are many approaches to treat cancer. You know, traditionally, chemotherapy has been the primary method, and this is, you know, has been for many years. And this was a huge breakthrough. But, you know, it kills all cells. But the reason it works for cancer is that the fast-growing ones die faster. Another way we tackle cancer is, you know, we have a drug called Avastin. There are other drugs that target the same pathway, but they basically block the tumor's blood supply. You know, I've talked about several drugs already that block the signals that make tumor cells grow. These are those targeted agents. And we can also give the immune system a boost so it can kill tumor cells. And this Tecentriq drug that I mentioned before actually masks those PD-L1 stop signs on the tumor and allow the immune system to act. And then finally, you know, we can modify the tumor microenvironment to make it less hospitable for tumor cells. And there's a lot of research going on at Genentech as well as throughout the field to try to figure out how to do this. So I just want to give a few examples of how we're tackling this tumor microenvironment characterization. Here's an example from a Genentech study of ovarian cancer, where we used digital pathology to quantitate and localize T cells in tumors. By analyzing different tumor sections and using algorithms to find different types of immune and tumor cells, we can say whether it's infiltrated, so all these dark spots are CD8 T cells coming into the tumor. Excluded, these are cases where the T cells are present, but they're not inside the tumor. They're around the periphery, and what we call deserts, where there are just very few immune cells to be found. So then, based on those digital pathology calls, we can bin them into each of those three immunophenotypes, and then combine with transcriptome data that we have to look at gene expression, and ultimately come up with tumor immune phenotype gene expression profiles that can differentiate these. So we derived a molecular classifier that can actually predict these different phenotypes without imaging at all, just through gene expression. And then we show that this molecular subtyping is also predictive of response when you treat with agents that target the immune system. So, imaging gets more and more sophisticated, and I think you'll probably see some great examples of this throughout the symposium, but this is an example of spatial multiplex imaging to characterize the tumor microenvironment. This is a study looking at 36 different proteins in triple-negative breast cancer samples out of Michelangelo's lab at Stanford, and they used automated image analysis to identify all cells and assign an identity to them based on the proteins that they're expressing. So you can see all the different cell types here. And then you can use spatial statistics to explore relationships between the cell types and the tumor and its environment. And they've done that in this figure here on the bottom right to show that there are different neighborhoods in the tumor that are enriched for different groups of cells. And I think this is a really promising way to start really understanding the biology of the tumor microenvironment in detail. So again, single-cell genomics has become increasingly important to our characterization of cancer. This is an example from a team at Genentech that profiled pancreatic tumors from mice and humans to assess the cellular diversity of fibroblasts in the tumor microenvironment. Here is their experiment on 20 mice. Here is their experiment on 22 human tumors. And using clustering techniques, in this case TISNE, in this case UMAP, you can identify different cell clusters based on their expression phenotypes and assign them into different cell types. And then focusing on this group here of fibroblasts, cluster 5 in humans, clusters 3 and 4 in, 3, 4, and then these tumor-associated fibroblasts here. They were able to go back and look in some of our clinical studies, for example, this bladder study here and this phase 1 study in solid tumors, to show that patients who had lower levels of these CAFs, these cancer-associated fibroblasts, actually respond better. This is a way that you can really get a detailed understanding of the role of different cell types. So just to start wrapping up here, it's our objective at Genentech to shape the future of cancer care. So since this is a grand challenge symposium, this is my grand challenge to you. Defeat cancer by matching every patient to a curative treatment regimen. This is going to be really hard, but this is really our ultimate goal. So I have to admit, I didn't have a lot of familiarity with the IEEE before being invited to speak at the symposium, but this is the mission statement of the IEEE. And, I really find this quite inspiring. And if the mission is to advance technology for the benefit of humanity, then, I'm hoping some of you will think about applying your skills and talents to, addressing this really huge challenge. I just want to point out that we've reached a convergence in the biological sciences between our understanding of biology, but also the computational methods and data sets that have become available to assess it. So around cancer biology, we have huge integrated data sets. We have deep knowledge of biological networks, a real explosion in machine learning and advanced analytics, plus the advent of massive throughput technology and powerful computation to enable all of this. And we really see this working with us in a human-machine partnership. This is how we look at the application of big data and advanced analytics at Genentech, is that we really want to use all of this to enable us to make insights and to solve these big problems in cancer. So with that, I will thank you and take questions.
Wow, thank you, Craig. That was wonderful. That was very inspiring, and I really like your ending too. The grand challenge. That was great. I guess one question I have just to start, we have a few that have come up, is that it is a daunting task, as you point out, to get to that personalized medicine concept. And for your estimation, do you think that's even possible, or is it more bucketed therapies, or how do you view this going forward?
Yeah, it's a great question. I think because of some of those evolutionary issues that I raised, it's going to be hard to beat evolution, and I think cancer is always going to try to find a way to outrun whatever we throw at it. So I think the future of cancer care probably is, how do we monitor patients closely over time and adapt treatment on the fly to, basically stay a step ahead of the tumor. So, there are some really nice methods that have been, that have been brought into the clinical care around monitoring using circulating tumor DNA, for example. And it may be that, if those become routine, that we could really see resistance mutations coming up before a clinical growth of the cancer. And, if we can make treatment decisions, then we can kind of maintain it at a low chronic level before it gets the chance to grow or metastasize.
Mm-hmm. We have a question from Shankar, from our past president, actually. Shankar, you have a question for Craig?
Yeah, can you hear me? Thank you very much for really an exciting talk. I enjoyed it very much. I like the notion of looking at the tumor microenvironment. The question for you is, there's a dynamic immune system remodeling that's going on at the epigenetic level, which is responsible for changes in the immune cell types. So is Genentech focusing on that? And follow-up question to that is, that gamma delta T cells are depleted significantly in the tumor microenvironment. Are you focusing on trying to address that issue of restoring the gamma delta T cell population?
What I can say is we have a large and growing cancer immunotherapy group, both in research and in development. And, we can't seem to bring immunologists in to fast enough to keep up with it. We are tackling it from many different angles. A lot of it obviously is centered around T cell biology, but also manipulating other parts of the immune environment. So, for example, I mentioned fibroblasts, where we have a research program. I think you really have to explore that whole space and really try to understand how all those different parts come together to make that environment more hospitable for the tumor, and try to combine different kind of molecular tactics to make that happen. As far as the epigenetic reprogramming and kind of cell fate shifts go, that's a tough problem, and I think we all are quite aware of it. There are drugs out in the market that and in research that people have been looking at for a long time that try to manipulate epigenetic modifiers, but because their effects are so pleiotropic, we don't. It's really hard to kind of control that at a granular level. So I think that's one of the frontiers for us.
Thank you.
Ali, you had a question?
Yes, actually, kind of a, let's say, perhaps open-ended question. How do you see the probability of having a vaccine against cancer? Because say BioNTech, for example, saying, hey, they're working on a vaccine to really get rid of cancer for good in the next 10 years, hopefully. And when you look back into the history of cancer treatment, starting in the ancient Greek or Rome, until today, I mean, there's been always a desire to get rid of cancer. How do you see the likelihood of a vaccine against cancer? And the reason I'm asking is because the genotype and the phenotype of cancer is so diverse. Cancer is a personalized disease.
Yeah, it's a good question, and we have a partnership with BioNTech to develop cancer vaccines, and the thought behind that is that we can identify mutations in patients that produce aberrant proteins that can be recognized by the immune system. So, we have the technology to do this. We can sequence the entire tumor of the patient, identify all of their mutant proteins, and then do some further work using some really interesting AI approaches to predict which of those will actually form an immunogenic protein that the immune system can recognize. So, this is really the foundation of the program. And, BioNTech has been manufacturing vaccines for patients, and we're running clinical trials right now to see if it really works. So, I think it's I think it's super promising. There are other companies in this space as well, like, Gritstone and Neon, who are going after the same idea, and I think it's super exciting. And I worry about evolution there too, but I think it's certainly a very promising approach that we're very interested in.
Fabulous. Thank you very much, Greg. Thanks for joining us. Great talk. If it's okay, then I'd like to introduce our.
Can I make one comment?
Yes, ma'am.
Yeah.
Okay, Craig, exceptional, highly inspiring, very interesting talk. I also appreciate that you put the one slide about the mission of the IEEE. At the opening remark, as we stated, the mission of IEEE Engineering Medicine and Biology also is to foster and promote global biomedical engineering innovations, research and education. And we look forward to be in touch with you and other colleagues, and you will be hearing from us. And we would like you to join also IEEE EMBS family as well.
All right, it'd be my pleasure. And yes, feel free to reach out. I think there's a lot to be discovered at this convergence.
A lot of synergies that are possible. I agree. Thank you. Thanks, Craig. Wonderful talk.
Thank you very much. Appreciate it. All right. So I'd like to introduce our next speaker then. Our next speaker is Kamali Madali. She will be talking about precision medicine and cancer and how technology barriers and breakthroughs can help to serve community cancer patients. And I've met Kamala a few years ago. She's a great person and very knowledgeable about across the whole continuum from pharma to diagnostics. And I'd like to just have Kamala on stage now.
Hi, everyone. Can you hear me?
Yes.
Awesome.
Good to see you.
Awesome. Good morning, everyone. Good evening, good afternoon. Let me share my screen. Please confirm if you can see my screen.
Yes, it's up and running.
And then let me just make sure. And once again, this is Kamala Madali. I'm calling from the beautiful city of Philadelphia in the East Coast. And the background is a beautiful vacation memory from Russia 2 years ago. So, and also really, it's an honor to really represent IEEE. Really, it reminds me of one of the prestige institutions in India, IIT, which is like the Harvard of the Asia. So it's an honor. And once again, Apologies if you have, if you hear any stuttered speech from me, because I'm a rare disease patient battling with multiple sclerosis and Parkinson's disease-like signs. This is my 9th year, and I sometimes have stuttered speech. Apologies, okay?
And Kamala, do you want to put your presentation into presentation mode so that we can use the full screen?
Absolutely, slideshow, right? Okay.
Yeah, thank you.
From beginning, okay, good. Okay. I just want to take a moment and really thank IEEE really for the mission and the vision, and also one of the questions that came, one of the first questions when Dr. Raimond has given a really prestigious and elite talk was, what's the future of medicine and pharmacy, or even engineering or technology? And if students should study AI or data sciences. Well, if a scientist like me can daringly love Mickey Mouse, and I put a photo here, as you could see, yes, be a learner and be a change maker and be a part of the change. Just a preview here, first 30 seconds, I'm a veterinarian in the human medicine world. I'm a scientist and a clinician in the life sciences world. I'm a non-engineer in the AI world. In my path to be a change maker in human health, I'm a non-MBA in the business world. I'm a business strategist. And I'm a shaman in the world of collaboration between men and women, and an empathetic patient champion because I'm a patient, and I'm a patient who's looking for a diagnosis. Imagine all those patients who have a diagnosis. And yes, a curse is a blessing in a disguise, but being diagnosed with a certain condition is a better situation than undiagnosed, where my battle is and my quest is. And a lifelong learner, teacher, and a mentor, just like Stephen Hawking and Michael J. Fox are inspiring and have inspired the world. And the power of hope that is driven by science and innovation and technology that we are all sharing, and everyone has been sharing since yesterday, is amazing. And let us not lose that impact. Be driven by the impact that we actually contributing to humanity, starting with ourselves. Thank you. And the talk will focus on advancements in precision medicine, overview of various therapies and diagnostics, and also investigational therapies, which is ongoing, sort of a pipeline of drugs and the relevant clinical trials. But also a special focus will be on clinical trials in precision medicine, and what are the challenges, not only in adoption of trials, but also adoption of diagnostics and treatments. Because a test is defining in oncology as, I think it's Craig from Genentech, a beautiful talk, Craig, by the way, thank you, who has laid out really complexity and the enigmatic, right? Where between, in relation to oncology, we are still learning a lot about cancer, and obviously neuroscience, my personal battle, trust me, we're not even near 10% of understanding. That's where I think neuroscience also will go with the footsteps of oncology. And what's the role of AI? That's where technology comes in. In adoption of innovation, you will see the challenges with all the advancements that are happening. The biggest challenge is, how can you help as many patients as possible? Because it's the type of complex information and the volume of information and case studies and prospects. Once again, a reminder, whose life? Why are we? Why are we in the field of healthcare? How? And what's the true north? Especially from an oncology perspective, our true north, any company. It can be a pharma company, it can be a hospital, it can be a lab, it can be a health tech company, it can be an imaging company, it can be a data science or informatics company. We are here to save lives. And here in this particular talk, I'm going to talk about AI being the warrior in this fight against artificial intelligence, but I'm coming up with a new name. It's an audacious intelligence and augmenting intelligence. Let's imagine a patient by name Gail. This is not a real patient. It's so basically, let's go through the journey and put our shoes into the shoes of Gail's life, who is actually diagnosed as a cancer patient. And once Gail starts interacting with an oncologist, with a health system or a hospital. These are the various personas or people that she will be dealing with. Pathologist, right? First tumor sample to understand the diagnosis. Oncologist, it can be surgical oncologist, various roles. Clinical research coordinator, what are the options available? If treatment is not an option, what trials, all of those, and a principal investigator. And this slide here explains about various steps or, through her patient's journey, Gail's patient journey, wherein an initial biopsy is taken, as you see on the left-hand side, path report, obviously the relevant sort of, downstream sort of, intentions, right? Why a path report is to understand the diagnosis. And then obviously Gail also has some moderate psoriasis, which a lot of cancer patients, that's the reality. They're also battling with other immune inflammatory disorders. And then obviously her first line of therapy commences. Then she goes through PD-L1. PD-L1 is one of the immunohistochemistry markers, right? It's a pathology endpoint. And you all know, right, in oncology, there is no single test. It's a battery of tests and multitude of tests at several time points. And then in 3 to 4 months, in the scenario of Gail, the condition relapses, and again, you see disease progression. And then obviously Gail meets her oncologist to discuss treatment options. And she's now trying to see if there can be a research option. And obviously the oncologist has reviewed and there is no matching approved treatment. So they have to go to see if she can be eligible for a trial. And the clinical research coordinator would actually is the one who would do the chart reviews, which is treatment history. And then you will see in the upcoming slides, what are the challenges in identifying a trial for a patient when there are no treatments. Before we go there, let's take a moment and appreciate how precision medicine, what Craig has alluded. I will not go through about the science behind the cancer, but overall precision medicine means right patient at the right time with the right treatment, which is also actually defined by right test. Nowadays, 58% of the industry's pipeline is driven by the type of test, which is actually matching as per the particular patient's biology. That's why not every drug will work for every patient. So you have to go through that individual level of assessment based on their genomics, proteomics. Now epigenetics is coming into play, lifestyle genetics. Oh my God, oncology is becoming more and more complex. But overall, 58% of the oncology medicines launched between 2015 and 2019 in the United States, especially these past 5 years, are loaded with pharmacogenomic biomarkers, which actually makes it more complex because test is defining, the fate of the treatment and the trial. So adopting the test itself, with my prior experience at Quintiles, IQVIA, Quest Diagnostics, everywhere, still the community is far away from adopting many of these tests. So across all these treatments, as you could see, this slide, which is put together by IQVIA Institute. So it's a very nice learning institute that IQVIA, which is a merger of Quintiles and IMS, has put together. All the various approvals that have happened, it's phenomenal. The advancements in science and innovation and how you see by indication in this circle of hope, I call it as, wherein various drugs have been approved. Obviously, one of the blockbuster drugs many of you might have heard, Keytruda. Keytruda has really made a huge difference, and many other drugs, from Roche, Genentech, AstraZeneca, Novartis, everywhere. Compliments go to every one of us who are involved directly and indirectly. And if you notice, the biomarker testing, which is predictive biomarker testing, understanding the biology of the drug, the biology of the patient, has consistently and steadily increased year by year, that these medications and treatments that are approved really are biomarker based, and more are increasing year by year as per the stats shown here. And then if you drill down, with a special focus on solid tumors, where you see in the center of this slide, There are multiple indications by phase, as you see, phase 1, phase 2, and phase 3, color-coded by blue, dark blue, and green, and also by pre-registration. As you could see, there are several biomarker-based treatments. And then the extreme right, you could see the type of genes, various genes that really would define access to those therapies and the biomarker targets that these therapies were designed against. Well, that's great. Until the research is happening, that's great. But then, when it comes to the trial design, that's when the complexity and the drama happens. As you could see here, it is contributing to complexity of trial designs. Trials are no more just as simple as, they were 10, 15 years ago. Biomarkers are now incorporated very extensively, as you have seen in the previous slides. And this particular slide, as you can see in the right-hand side, look at the type of biomarkers that you have to look at in technologies from IHC, FISH, flow, PCR, next-gen sequencing, traditional PCR, nanostring technology. So technology has a strong play in understanding the biology of the patient and the biology of the tumor, systemic biology, local biology of the tumor, and also the mechanism of action of the drug. So overall, there are there are many basket and umbrella studies that would complicate the patient identification for care teams. Because let's not forget, for the trial to happen, we are going to the hospitals. The hospitals, the physician or the KOL and their clinical research staff is out there. They are not up to speed with the evolution of these sort of complex trial designs. That's the reason why AI has a lot of play into adopting trials and treatments, which you will see in the upcoming slides. And the struggle, as you see, while we are spending 5.8 billion median per drug from big pharma, 2.6 billion for taking a new drug to market, and 8 million each day that a trial delays a blockbuster drug launch. 25% of trials never reach full enrollment. 41% delays, and 5% of qualified patients only are enrolling in a trial. We're talking about a very big problem here in the industry about how do we really get drugs faster to the market. We follow the same conventional procedures and to be more clear, conservative procedures. While technology in cancer biology is advancing so much, why are we not adopting using right technology, today's technology, leveraging trials and treatments as a care option? And once again, this slide speaks for the problem. 11% of sites, clinical trial sites, fail to enroll even a single patient. 37% do not even meet their enrollment goals. 40% of surveyed adults don't understand clinical trials. And 2% of surveyed adults say they consider participating only after they understand what a clinical trial is. There is a clear need for patient awareness, patient education, physician awareness, physician education, provider awareness, provider education, payer awareness, payer education. There's a grassroots initiative very much necessary at every level, irrespective of the expertise involved in oncology care. Once again, let's drill down into the details. Why patient recruitment suffers in precision oncology? As you have seen in earlier slides, and also what Craig has alluded from Genentech. Sorry, Craig, I'm taking you as a role model example because of the science that you have discussed in your presentation, which was stellar, is basically you're dealing with massive data points. As I said, you have to take pathology, you have to take testing, liquid biopsy, immunohistochemistry, all the various biomarkers. It might be at one time point and several time points in a patient's journey. So imagine the health systems out there are dealing with these massive data, but not everything is sitting in electronic medical records. Many of it is sitting in pathology, lab information management systems, and genomics never sits in any of these sources. So you're dealing with various data sources, which can be in an EMR, which can be in a PDF, which can be on a post-it scarily in some scenarios, or Excel sheets. But then when you look at a complex clinical trial protocol, or any clinical trial protocol, it is loaded with inclusion-exclusion criteria, starting to imaging, CT scanning, treatment history, prior treatment history, if they've seen another oncologist. So you have to really conglomerate all of these sources and then match the patient for a particular trial. So there is a big gap how you really integrate all these massive data sets, complex and siloed, and there is limited connectivity and interoperability because many of the systems don't talk to each other because of the antiquated infrastructure, and then ineffective manual tools, right? As I said, sticky notes, and no automated population of data fields, and lack of community oncology coordination. One of the biggest challenges in oncology is 70 to 80% of the cancer patients are in the community setting. They don't have access to big academic medical centers or top NCI cancer centers. So we have to be very realistic how we leverage novel treatments or investigational treatments or trials to the patient or to the common man. Once again, if you put Gail's journey here, several scenarios will be there as you see, first line of treatment, and then the patient is not responding. They might be considered for second line of treatment, but many scenarios, they might not be eligible based on their unique biology of the tumor. So you have to look for this window of trial. Many cases in real life scenario, they have no choice. They are just extended either on chemotherapy or they're put on anything that the oncologist would think would be appropriate, which is unfair with the advancements that are happening, that you really have a chance to really adopt technology in clinical decision making and also in matching the relevant clinical trials. And one of the biggest problems, as you see on the slide, is that window of opportunity, right? When the patient stopped responding to the first line of treatment or second line of treatment, which you would hear in all these trials and also approved therapies, right? Is this particular clinical research coordinator is going through all these data sources to understand, once again, what best can we do for the patient. And on average, it takes 11.8 hours based on various sort of studies that are done. Overall, it's taking few hours per patient, by a clinical research coordinator. And once again, these are not dedicated or committed resources. They are physician navigators or nurse navigators or full-time nurses who are trying to do this ad hoc work. So once again, these are not, so that's a big sort of a gap as well, and a challenge wherein these are not dedicated resources to leverage additional options in the cancer care continuum. So now let's understand how technology can automate the identification and the screening process of the patient's eligibility for a clinical trial. So I represent Deep Lens. So one of the role models I just want to take here is on behalf of Deep Lens. I thank Deep Lens, for letting me share this slide, wherein, as you see here, the AI solution over the cloud, either on Azure or on Microsoft Azure or AWS, Amazon's web services, is deployed in hospitals, in networks, and also cancer centers. It would integrate with any electronic medical record system, any pathology, any lab system, any genomic lab, and push all the patient data, in a HIPAA-compliant manner to the Viper cloud within the hospital system. And then using API, NLP, natural language processing, and all of the information is taken, centrally integrated, and then structured, and then put into the web-based solution to identify patients for a relevant clinical trial that is either ongoing at that particular site, or new trials will be brought to that site based on the patient profiles that the AI solution is actually, identifying. Overall, the goal is more enrolled patients, higher patient retention, higher physician satisfaction, and better operational efficiency. And obviously, new revenue opportunities for the health systems as well, in addition to treatment of patients, but by leveraging trials. And very important aspect of machine learning and data integration here is all these integrations, as I said, irrespective of who the ultimate party is it Epic, is it Cerner, is it Medent, is it iKnowMed, is it OncoEMR, or is it molecular test integrations? These are all various genomic sort of players that would leverage next-gen sequencing or liquid biopsy or IHC testing. So it doesn't matter, it's an universal data source integration model that AI is leveraging. And then providing dashboards, wherein why a site is able to enroll, why a site is not able to enroll, and why some patients consented, why they could not consent, what are the various factors. They can be pre-built, they can be as we are going through, and what are the scientific factors, clinical factors, medical, operational, regulatory, lifestyle factors that can be captured, which is wonderful information to be given to pharma companies and also to the health system, right? This is the voice of your patient. And that's where I think AI also has a strong role in addressing health disparity issues, as you're hearing throughout. Because of the COVID scenario, you might have heard a lot about credibility. Are we really leveraging the innovation from treatments and trials and science and innovation perspective to as many patients or to every patient possible? So you can monitor all the outreach initiatives as well, and then also assess why the outcomes are poor or better. And going back to Gail's journey, by leveraging technology, as you could see, how technology can really help by taking the information from our biopsy, from our path report, first line, and then additional testing. All of this information, the AI solution follows the patient longitudinally. And then it would alert, because it's a lot of integration, automatic integration, taking away the human sort of burden. Once again, being realistic, not everything can be taken away. There is a lot of information sometimes as clinician notes, charts that have to be reviewed manually, which technology companies have realized that you need an on-site dedicated person. And there is an on-site person who is dedicated to help the health systems adopt the technology and also help them identify patients and then flag within 2 weeks to identify the right trial, the right patient. And this is how I think we can empower a lot of community oncologists out there, because they don't want to lose the patient. That's another biggest challenge we're talking about, right? They don't want to lose the patient to a bigger cancer center. Technology has really a strong play in becoming a catalyst for adopting science and innovation. And community oncologists, can access real time, can understand what options they can give. And also, once again, AI is like a teacher, right? It's the machine learning component is really helping the community oncologists, not only with trials, there are other AI solutions which are helping. The moment you put from day zero, the patient's information, it's guiding NCCN guidelines, treatments, emerging tests that needs to be done, right? The various biomarkers, then it will guide what sort of treatments are available based on the emerging tests, and then what sort of trials that can be leveraged through a Viper-like solution. Overall, clinical research in cancer care cannot be an option. It is a responsibility for us on behalf of the industry. And to everyone out there, collaboration is a higher source of innovation, as we are seeing through AI in medicine and healthcare. So stay positive. You must be the change that you wish to see in the world. I loved He-Man when I was, I think I was a 6th grader in India. At the same time, I grew up, as you see in this photo, one of the idols that we worship, we call her as Durga on the lion. I'm like, oh, okay. Anyone can be a warrior. And it's all within you. Go, interact, network, mentor, and be a mentee. Thank you very much.
Okay. Thank you, Kamala. Thank you very much for your talk. That was really great. And I like the ending with the He-Man and the Battle Cat. I'm not a big fan of that. No, not sure why this disappeared. But first of all, like what I see as a great takeaway is also how Gail's journey can turn the very complex chemotherapy into more immersive experience for the patient, because I think it's very difficult even just to stick to your medication. So I think that's a great tool you are working on. So congrats on that. And then Colin, my co-chair, has a question, I think, who wants to address.
Yeah, well, I have 2 questions. First, it really plays off of what Craig, he had a grand challenge for us as a society, IEEE. So I was curious, Kamala, what would be your grand challenge to us? When you look out there and say, hey, you know what, this is a big problem area, which you have described. How can we as a society make a difference here? So I think that would be my first question.
Okay, wonderful question. My personal journey and professional journey, how it's intertwined. We need to raise the next generation of scientists in the medical world. Really, everyone has to be a clinician scientist, not just a clinician and not just a scientist. So, and foster more collaboration, as I said, because it cannot be standalone. An engineer cannot be standalone, a doctor cannot be. it's all interlinked. We have a mutual goal to address.
Mm-hmm. Wonderful. And my second question is really more sort of technical nature of that, because we learned, from Craig's talk about the intrinsic tumor heterogeneity, the high degree mutations in tumors and such, and they're being characterized now through next-gen sequencing. And so, but as you know that, I think you talked to us in your talk simply that about the confounding nature that because of that heterogeneity, that it's hard to enrich for patients for a particular test or for a particular drug to be tested because of that confounding nature, right? It's hard to be able to get that select group of patients that can be best responders to the drug and not have confounding results. So it's kind of curious that do you feel that the current measurement tools are adequate and they're prevalent enough, because you talk about community oncologists, which is a great idea, but is there enough out there in the community in terms of testing that can be done to be able to select for those patients that could be potentially best responders to the drug under test, then ultimately give better quality clinical trials that can give us better guidance as to that more personalized treatment?
Very good question. Especially as I alluded in the past 5 years, how many number of therapies have been really, biomarker based, right? And more genomic based. So one of the biggest learnings that we are seeing based on the lessons learned from the commercial launches, right? How many of them are adopting out there? There's a big gap. Basic gap is what is next-gen sequencing? What is a biomarker? There are many of them who have actually no clue. Really, that's why I said patient awareness. Mm-hmm. Patients have to be also be empowered with the knowledge. One of my roles in the industry is actually I serve as a board member on a few of the cancer patient advocacies, and also on one of the digital health companies, which was started by cancer patients and who are actually educating patients by really, by hosting more scientifically sort of, and also a common way of, man's way of understanding what can biomarker do for a scenario and situation. And on top of that, the scary part is even the oncologists, many of them have no clue because they're far away from innovation, nothing wrong. Their lives are so busy. That's why technology, everybody would have an EMR, right? An electronic medical record system. EMRs are now becoming mentors in adopting. So they're like, I would call them as precision EMRs, meaning you have to really, they cannot be just plug and play model, right? They're also learners. There's a huge machine learning component coming into these EMRs, wherein it is actually guiding the oncologist. Have you done this testing? Are you aware of this NCCN guided testing? That's the future to go. And it's not going to happen overnight, right? So it's basically baby steps, but technology has a strong role to play. Number one, bringing awareness of the testing before you even talk about the treatment. First thing is testing. And while I was at Cost Diagnostics, it's a public report. Not even 30% of the people knew about BRCA testing in breast cancer, one of the most prevalent genes, right? So you're talking about a big gap in awareness and education at every level. And technology has a strong role to play, not only as a reminder, but also how can you blend into their workflow process? That's why I think when any of these systems, where the hospital or community are accessing, technology has to be those responsible mentors as well that are reminding and that are actually coaching the oncologists about the latest guidelines, testing and treatment perspective.
Mm-hmm. Great, thank you.
You're welcome.
Okay, let's see, any more questions from our audience? Or, yeah, probably not. Then I think the stage is yours, Colin. You can make the introduction of our next speaker.
Great, thanks very much, Kamala. Wonderful.
You're very welcome.
Thanks, Kamala.
So it's my pleasure to next introduce Dr. Curtis Harris from the NIH. Dr. Harris received his MD from Kansas University School of Medicine and completed his clinical training at the University of California, Los Angeles, and at the NCI. He has held positions of increasing responsibility at the NCI and is also an adjunct professor of oncology at Georgetown University School of Medicine. Dr. Harris has received numerous honors throughout his distinguished career, including the Distinguished Service Medal. That's the highest honor of the U.S. Public Health Service. NCI Outstanding Mentor Award in 2007 and 2013. A PhD, an honorary PhD from Nippon University School of Medicine in 2013. And in 2009, he was awarded the AACR Princess Takamatsu Award. And in 2014, he was awarded the ILCA Nelson Fatso Award and the AACR American Cancer Society Award for Research Excellence in Cancer Epidemiology and Prevention. In 2016, he was awarded the Distinguished Medical Alumnus Award from Kansas City School of Medicine. And in 2020, he was awarded the Environmental Mutagenesis and Genome Society Annual Award. Dr. Harris has published more than 500 journal articles, 100 book chapters, and edited 10 books, and holds more than 30 patents owned by the US government. He also serves as editor-in-chief for the journal Carcinogenesis, and has held or currently holds elected offices in college societies and nonprofit foundations, including the American Association of Cancer Research, the International Society of Differentiation, the Keystone Symposium on Molecular and Cellular Biology, and the Aspen Cancer Conference. So it's our great pleasure to have Dr. Harris here today. And he'll be speaking to us on precision medicine of lung cancer. Welcome, Dr. Harris.
Thank you very much. Thank you for a very long introduction. I am going to talk about precision medicine of lung cancer. And the kind of traditional medicine that I learned, and let me get my pointer here. There we go. I thought. All right, it is to take a patient's history in terms of lifestyle, medical, and family history, determine signs and symptoms, and do standard laboratory tests and imaging. Precision medicine is different in the sense that it's multi-layered, it's individual centric, and it's interconnected. And it utilizes various omics combined with the clinical and epidemiological information and the traditional medicine. And this leads to the information column. And this is just another view of that, in which the knowledge network gives one a new molecular taxonomic classification in individual patients to improve clinical medicine. And there's a feedback loop to the information commons. Secondly, this leads to informing biomedical research and informed mechanistic studies, which again enriches the information commons. And we added guide to prevention research. So smoking, you think of relationship to lung cancer. Indeed, that's the case, in which both males and females have a high incidence of lung cancer. Males are starting to go down, and actually females are starting to go down too. But in 2014, the Surgeon General report made a catalog of deaths associated with smoking, and not only for lung cancer, but for cardiovascular disease and so on and so forth over a 50-year period. So smoking has led to more than 20 million people dying prematurely related to their smoking addiction. You can smoke cigarettes directly, but you can also inhale someone else's smoke. And this is a watercolor by Professor Hirayama, who presented at this particular meeting a long time ago, in 1981, the first evidence that passive smoking increased risk of lung cancer. And at that time, I was in the audience, and probably half of these scientists and physicians did not believe him. But he proved to be correct, and there's well over 100 papers, scientific papers now, linking passive smoking with lung cancer risk. And since this was in Greece, and I like these kinds of quotations, some men have constitutions that are like wooded mountains running with springs, others like those of poor soil and little water. Still others like land rich in pastures and marshes, and yet others like the bare, dry earth of the plain. Hippocrates. Well, about 30 years later, we decided to ask the question whether or not, and this is a hypothesis, that childhood exposure to secondhand smoke and genetic alterations in innate immunity would increase lung cancer risk in never smokers. Now, we had to wait almost 30 years because women didn't start smoking until the Second World War. So, and then, of course, started having children. So Susan, who conducted this study with our colleagues, and the result was that actually parental secondhand smoke exposure during childhood is associated with dose-dependent increase in lung cancer risk among never smokers in 2 separate cohorts. This was especially true in individuals who had a mannose-binding lectin-2 haplotype in their DNA, which is associated with hyperactive innate immune system. And somewhat surprising, these never smokers develop lung cancer at an earlier age than one would normally suspect. So let's move on now to the exosome. And this was coined by Chris Wild, who was at that time the director of the International Agency for Research on Cancer. So it includes not only the external environment, such as tobacco smoke, infections, diet, and so on and so forth, but things in the internal environment that are directly related to this. Obesity, and I'll say something about that in a moment, and chronic inflammation. Of course, the cancer genome and epigenome plays a role in cancer susceptibility and driving cancer and cancer genes. And there are certain, this can lead to certain types of therapy that are directed to specific mutations in cancer cells in their DNA, such as EGFR mutation. And this not only gives you an insight into therapies, but also understanding carcinogenesis. So all this together can lead to cancer biomarkers of risk and diagnosis and prognosis and cancer prevention and screening strategies that you just heard about. Now let's turn to inflammation. Chronic inflammation and infection can increase cancer risk. It can be inherited more than acquired, such as hemochromatosis and iron overload disease that leads to liver cancer, to chronic inflammatory disease, Crohn's and ulcerative colitis, and familiar pancreatitis. So President Carter, who's now about 100 years of age, his family, many of his family members died of pancreatitis and pancreatic cancer. He's still alive with actually melanoma and the normal pancreas. More frequently, it's acquired more, inherited by exposure and the effects of viruses, bacteria, and parasitic organisms, and of course, chemical, physical, and metabolic conditions, acid reflux, obesity, which I mentioned earlier, and smoking. So about 2 million human cancers per year are related to infection, and other causes of infection are many more cancers, and actually smoking, in addition to containing more than 60 chemical carcinogens, also is a potent inflammatory agent. Now, inflammatory cytokines are associated with lung cancer risk, diagnosis, and prognosis, and survival, I should say, also. And in prospective studies, we found that even 20 or 30 years prior to develop lung cancer, there's an increase in this cytokine, interleukin 8 and C-reactive protein. So they already have an activated immune system. And at time of diagnosis, one again finds an increase in their blood system of IL-8 and IL-6, both in Afro-Americans and European Americans in the United States. It's a study done in the United States. And IL-1 beta and interleukin-10 in African Americans. In terms of survival, there's a continued association with high levels of IL-6 and IL-8, this mannose-binding lectin haplotype, which I mentioned earlier, TNF-alpha, another pro-inflammatory agent, and two other cytokines, IL-10 and IL-12. I mentioned obesity also, and actually obesity is a chronic inflammatory disease, among other things. And in obesity, one finds adipocytes, and these adipocytes, as they divide, can become senescent and start producing senescence-associated secretory proteins, including a number of pro-inflammatory agents. And there are feedback loops that go back and forth between the adipocytes and, of course, these cytokines and pro-inflammatory agents are found in the blood. But more recently, it's been found that there are macrophages, which are in the body fat, which again form very potent feedback loops with the adipocytes and increase the inflammatory milieu. Genome, that's something that's probably most of you are familiar with. That's mutations in various genes that may predispose to developing of cancer. And this is just a bar graph of a variety of different genes that are mutated in a variety of different kinds of cancers, from ovarian cancer down to pancreatic cancer. And as you can see, that the highest frequent gene that's mutated is actually P53, a tumor suppressor gene that, as I'll mention, when it's mutated, loses function and gains function as an oncogene, a cancer-producing gene. Well, the discovery of P53 mutations linked to environmental carcinogen exposure, some people consider the beginning of molecular epidemiology. And this is just a catalog of some of the carcinogens that actually cause mutations in different tissues, such as aflatoxin and liver cancer. This is the kind of mutation that takes place in the DNA and G-to-T transversion. And there are hotspots in the genome, in this case in the P53 gene, that are associated with specific types of carcinogen exposure, such as aflatoxin. It's true for tobacco smoking and ultraviolet light and astrodolic acid, which is associated with pancreatic disease and, renal disease and renal cancer. Well, since those early studies, there's computational analysis of the entire genome sequencing has identified endogenous and exogenous induced mutations, including those caused by chemical physical carcinogens, inflammation, DNA repair, and defects, and also just with aging. So there are catalogs of signatures that have been developed, which are linked to different diseases, different types of cancer that are listed up here, and probable association here. And this is a very early study. There are now more than 100 signatures that have been linked to causes of cancer. And of course, I mentioned smoking earlier. So smoking increases the risk of lung cancer and also head and neck cancer. And it also contributes somewhat to even liver cancer. Now, those are associations. Can you develop some laboratory studies that support those associations? So this happens to be the mutational signature in lung cancers. And if you expose benzpyrene to animals that have been genetically modified so that they have the human P53 gene, you get more or less the same sort of signature. And for human cells exposed to benzpyrene, which is one of the 66 different chemical carcinogens found in tobacco smoke, it has the same sort of signature. The transcriptome and the which is the message RNA, and the epigenome, microRNA, and DNA methylation is another part of information commons. MicroRNAs were discovered in C. elegans, which is a small worm, and these small non-coding RNAs that are evolutionarily conserved and regulate gene expression were found by Victor and Gary in 1993, and protein output of hundreds of genes are repressed by each microRNA, destabilizing the message or increasing the extent, lessening, I should say, and inhibiting the translation of that message into proteins. It was a decade later when Carlo Croce discovered that in human cancers, that microRNAs are differently expressed, and that these microRNAs can predict risk, diagnosis, prognosis, and also therapeutic outcome. So this is just a diagram of the two main functions of microRNAs in inhibiting one, translation of the message into proteins, and the second is inhibiting the message RNA by causing it to degrade. So you no longer have it translated into proteins. But microRNAs can also bind to proteins and alter protein function, and also can bind to receptors, including receptors that are involved in inflammation that I've talked about before, IL-6 and TNF-alpha. So one of the early studies that we did actually with Croce was to analyze 6 different cancer types and determine the preponderance of microRNAs of specific types in those cancer types. At this time, there were only maybe 200 microRNAs that were known. Now there are close to 2,000. So that's just an advance that's been made over the last 14, 15 years. So out of these cancer types, miR-21 won the race. It was highest in all 6 cancer types, and these others were high in various other cancer types. So subsequent to this, miR-21 is both upregulated in 18 major cancer types, and is a biomarker of survival in 14. So these are some plots that's usually called for lung cancer, for colon, for pancreatic, for breast, and so on and so forth. And this is 100% survival, 0% survival, and this is a 5-year point. So low levels of miR-21 in lung cancers of varying types, better prognosis than if there are high levels. And that's true for all these various cancer types that are described here. So this also led to interest in the function of miR-21, how it's increased, and gene amplification is one of those, especially in lung cancer. Decreased transcriptional silencing can lead to an increase in miR-21. EGFR and KRAS, which can be mutated, both of these can be mutated in cancer, can lead to an increase in miR-21. And inflammation again, IL-6, IL-17, and interferon through STAT3 can increase miR-21. And different kinds of DNA damage from free radicals or ionizing radiation. So how they function, they inhibit the translation of the messages into this series of proteins that are related to cancer growth, and so that they no longer inhibit cancer growth or invasion of metastases, and so on and so forth. And as I said, miR-21 can also lead to inflammation itself, so there's actually a feedback loop here, and cachexia. So cachexia is a devastating side effect of cancer. And about 30 to 40% of the people who get cachexia will die before they die of cancer. And this is the cancerous tumor itself. And this leads to a loss in muscle mass and in fat. So the body just essentially melts away. It's a devastating disease. Now, one can do analysis in different populations, such as Japan, of early stage lung cancer, stage 1A and 1B. It's 3 centimeters or less, or 5 centimeters or less. And then again, these are survival, 100% survival, 0% 5-year. So if the MIR21 plus these 1, 2, 3, 4, 5 gene expression proteins, if those are also expressed at a high level, a poor prognosis. So this is the 4 genes, this is MIR21. If you put them together, they give you actually a truer result of the risk, or the prognosis, I should say. This is a companion study in the US and Norway. Again, 4 gene signature, poor prognosis, and if it's high, and MIR21 if it's high. And if only one of those is, if they're both low, very good prognosis. If one is high, if they're both high, even much worse prognosis. So why might this be the case? And this is a diagram of non-coding RNA and coding RNA. And you combine them, you get what I just showed you previously. If they're both risk is for prognosis is low, that's good, a good result. If either one is high, poor prognosis and survival, and if both are high, very poor survival. So each of these biomarkers, if they're in agreement, give you a good indication of what's going on. If one is misclassifies some of the patients, then the other may not, but may misclassify other patients. So the combination is better than one or the other because there's less misclassification. All right, let's more or less in with the metabolome. So tumor metabolism leads to various metabolites, and the study of this is called the metabolome. These are my 2 colleagues who test the hypothesis using mass spec analysis of urine, serum, and plasma. And they used unbiased metabolomics. There are close to 2,000 proteins now that you can detect in serum or urine that will discover biomarkers associated with risk, diagnosis, prognosis, and therapeutic outcome of lung cancer. So these are all, can be all used as biomarkers of risk, diagnosis, prognosis, and also therapeutic outcome. So out of that large number of proteins and using random forest, which is a bioinformatic approach to identify those metabolites that are, in this case, the most related to disease. There are 4 different metabolites: greeted riboside, which we now have discovered exactly what it is and how it's formed, N-acetyl muriatic acid, corticosulfate, and a peak at that when we originally did the studies at 561, which now we have actually identified the compound that leads to this peak. So again, if only one is high, it's high. This is the prognosis of this early stage lung cancer. If 2 is high, if 3, if all 4 are high, then you have the worst prognosis. Kaplan-Meier plots again, 100% survival, 0%, and this is 5 years. So the combination gives you a better predictive analysis than just 1 or 2 or 3. In this case, 4 is better than all of that. Now, there are other ways of detecting early stage lung cancer. And one of those has been developed in recent years called low dose computed tomographic screening, LDCT. And the traditional way of screening was by X-ray, which was not very good because it only detected large tumors. But low-dose CT actually reduced, in this initial study that was in the New England Journal, mortality by 20%. There was another large study in Europe that gave the same result. Now, why might this be the case? Well, low-dose CT actually can detect these small tumors, even those that are 3 centimeters in size or even smaller. And when you do surgery on these, you have a much higher probability of a cure. However, about 25% of the individuals with stage 1 lung cancer will reoccur and die of the disease. So how might one identify those individuals who would be in this 25% category? Again, using precision medicine is the strategy. You decrease the false positive rates, decrease financial cost, that's important, improve patient care, and guide mechanistic studies. So these very small tumors, again, both are removed by surgery, but some are low risk and some are at high risk. So the ones that are at high risk, we would like to offer adjuvant immunotherapy or adjuvant chemotherapy. In general, stage 1A, particularly surgery is the therapy, and there's just wait and see if there's a development of metastases, in which again 25% of the patients will occur. So, but if you could identify these individuals at the time of surgery, then you could have an opportunity to decrease their risk of recurrence. So this is some of my colleagues at the NCI and around the world that have contributed to the studies that I very briefly have described. And I want to thank you for your attention. I'm happy to entertain any questions.
Well, thank you, Dr. Harris. That was a wonderful presentation. Really, very, very interesting. So we do have a couple questions. So from the audience, Carlo Robles asks, could heritable diseases be explained as generational exposure to carcinogens?
The answer is contribute to that. So there could be heritable diseases that give you a hyperreactive immune system. I gave which that mannose-binding lectin type 2 haplotype increases the risk of passive smoking. And in some cases, heritable diseases have a poor DNA repair, and that can lead to an increased risk of various kinds of cancer, such as ultraviolet light. And then lastly, you can inherit mutations, for example, in the P53 gene, and lead to the Lee-Fraumeni syndrome, in which the offspring develop multiple types of cancer. And then there are many other examples of inheritable diseases that increase the risk of lung cancer. And in some cases, as I said, increase the risk associated with sunlight, for example, and the poor DNA repair.
Interesting. Ali, you have a question?
Yeah, so thank you, Curtis, for your great talk. I did enjoy it. I have a question is, when you look at chronic inflammation, obesity, and infection, how do you see to incentivize society to basically watch better out to avoid chronic inflammation, which is very often caused, not only, but to some extent also by nutrition? It's like, where do you see the prevention? Because on one hand, we have all the back end with cancer and all the complexities, but I think there could be so much being done on the nutrition side. What's your take on that?
Yeah, So to put it in very simple ways, BMIs, which is a measure of obesity, is associated with lots of different diseases, cardiovascular disease and cancer. And so maintaining a normal weight and diet, of course, can play a role. There are certain infectious diseases that you can get vaccinated against, and including hepatitis B, and for example. So there are ways that you can protect yourself and decrease your risk of developing a cancer of various types.
Interesting. I have a question. It was really interesting. You laid out all the different layers of
Omics.
information
Yeah.
that exists in our bodies in different levels of regulation and such. I know there's been efforts over the years to develop a framework to integrate all these pieces of information together, and both at across the different layers, but also in a dynamic way that to be able to be more predictive of the number of biological, physiological function in general. How far advanced is that from your perspective and the kind of work you're doing relative to a specific disease like lung cancer?
Yeah. So all the omics I mentioned, I didn't have time to talk about a lot of the data, of course, can be combined together. And there's, the external factors like smoking, but who among smokers is more likely to get cancer and at an earlier point? Well, it can be related to inflammation. It can be related to DNA methylation studies that we've done and others. It could be related to DNA repair. So the integration of all that is something that has been done over the last 10 or 15 years in a very primitive way, but with these large data sets that one can get now and large analysis, more sophisticated ways of AI and other strategies are being used and tried. So it's a very exciting area of research right now in cancer research. Some people call it bioinformatics. There's lots of different names. And it's putting together large pieces of data, and whether it's from the proteome or the epigenome together to better understand risk diagnosis and prognosis.
Mm-hmm. So in that vein, then, as we're asking each of our speakers, what is that, from your perspective, what is that grand challenge you'd put to the IEEE, to the engineering community, that from your perspective, what would be that challenge to our audience that from maybe perhaps a lung cancer perspective, from diagnostics or prediction, or perhaps from another area that is of interest to you?
Yeah. So an example of engineering that has helped lung cancer is low-dose CT, as I was just mentioning. Mm-hmm. So right now, that kind of approach is being refined. So and there are algorithms that are associated with better prognosis at identifying benign tumors versus malignant tumors. So this is another kind of technology that is contributing to human health, in this case, cancer and lung cancer. Understanding the content and the biological effects of exposure to various agents in the environment, or, for example, e-cigarettes, which are, again, mixtures of nicotine and all kinds of other things, which are starting to show two things. One, lung damage, and then secondly, kids that are using e-cigarettes tend to then have a higher propensity to start smoking cigarettes. So the tobacco industry, which funds most of the e-cigarettes, now has a way of getting more smokers. This is my editorial here. More smokers to smoke their cigarettes and to die of cancer and many other diseases. As I mentioned at the very beginning, 20 million people over a 50-year period, just in the United States, had died related to tobacco smoke.
Yes, indeed.
Colin, may I ask the last
Benton.
question?
Yes, please. Yeah.
Curtis, fantastic talk, highly inspiring. I really, really appreciate your joining us and giving the plenary talk on behalf of IEEE EMBS. My question about the MIR-21.
Yes.
It's MIR-21P is heavily involved with COVID-19, right?
Yes.
And so can you comment on because one or two slides you showed us MIR-21 is very important biomarker sort of to look at long-term the efficacy or the long-term effect of the influence of the lung cancer. And is it also we are talking about the same MIR-21 here, right?
Yeah, MIR-21 is, as I said, is a microRNA that was discovered associated with multiple cancer types and increases well, it actually decreases the body and the cells from delaying or preventing cancer. And so that's how it functions.
Okay, thank you.
Great. Well, thank you again, Dr. Harris, for joining us today. We appreciate your time. And Ali, you'll introduce our next speaker.
Yes.
Thank you very much.
I'd like to
Thank you.
Thank you. I'd like to introduce now our next speaker, Dr. Thomas Wilkins. And so I've met Thomas more than 10 years ago at the airport in Salzburg. He is a very visionary physician and has great models and is working on exciting precision medicine applications, also based on inflammation. Thomas, are you on stage?
Yes, can you hear me?
Yes, perfect.
Okay.
Okay. Do you want to share your screen?
Yes, I gave you the permission already. I should work.
No, you have to share your screen. Or do you have it handy, or do you want to resend to me?
It should work now. Does it?
No, it's not. Go to the green share button, share screen button.
Okay. I did. Hold on a second. I had to use the computer of my daughter, which is an Apple. I work in the Windows world, because in lockdown you can't get anything replaced. Just hold on a second. We might be gone for a second.
Is there anything we need to do at our end, Ali or Nicholas? I don't know if you can help us.
I think Dr. Wilkins, I think they're going to resign and see if it works.
Okay, all right, that sounds good.
Nick. Yes. How are you doing? Okay. We are trying to get Dr. Wilkins to speak, and it looks like we have some issues with the sharing. Can you join the panel 2 seconds? Dr. Wilkins. He's on the panel right now. You will see him.
I'm sorry, what was that?
He's on the panel. You should see him. Wilkins. Thomas is the name.
Can you hear me? Can you hear me
Yes,
now?
Thomas.
Yes,
Hello.
we hear you.
So
Thomas is the first name. Thomas.
Okay, he's on now. Thanks.
Okay, good. He's on, but he's okay.
Ah,
Good,
okay.
good. Thank you.
Yep.
Thank you, Nick.
There we go. Excellent.
Thomas, we can
Very
see
good.
your screen now. Awesome. Thanks.
okay, good. So thanks for the invitation and apologies for the delay. As I said, with the lockdown, it's a little bit difficult to get a new computer and I can't actually get it shipped where I am right now. So I'm going to talk about precision medicine as a data-starving new discipline or paradigm shift. And I can't avoid to comment on what I'm most concerned about in science right now is that we have several mainstream trends and scientists start fighting with each other with a language that I find most disturbing. And I think if we want to overcome this pandemic, we must return to a more neutral, logic, and respectful dialogue. My position is pretty clear. Everybody can follow me on LinkedIn. But I think the way we deal with each other, the wording and the bashing in media is just disgraceful and is harming science and the reputation. So now get back to precision medicine. This is me in the morning when I get up and I should go exercise. By the way, Ali said we met 10 years ago. That's more than 20 years ago, but he wants to hide his age. So, and when I get up in the morning, I basically go to the fridge in the future and the fridge knows me. He knows my personal molecular profile. I will allude on that a little bit later. And he will recommend the perfect diet. If you think this vision is just like a cartoon, actually, there are fridges that buy and go shopping and groceries for you already. There is a complete technological role model in Finland where they play with these tools. And this sketch actually was drawn after I had a meeting with GE before they sold the refrigerators to a Chinese company. So going back to data-rich sources and prevention. I'm coming from Munich, so I'm taking the car of BMW, which has today about 800 sensors. These 800 sensors generate terabyte of data. You have to imagine it's like you have a full bookshelf at home with data that are being generated just while you drive. And this, if you're hooked up to the internet, this is basically real-time, precise diagnostics, predictive analytics. It's the best treatment in real time, and you have reduced long-term damage. So now think about how many data you get when you have your physical exam. An ECG, maybe 20, 30 blood parameters. That's pretty much pathetic. So we are not really using the opportunities we have with molecular diagnostics as the previous speaker was already alluding to. And now you guys are data scientists, most of you, as much as I learned. It's a pretty almost trivial task right now. You need about 40 parameters for face recognition. And then go back to how we diagnose diseases. We have very few, very robust biomarkers like blood glucose that is we have nothing which is dynamic. Today with face recognition, you can actually recognize just parts of the face. And what we do basically to illustrate a little bit how poor diagnostics is, most of you use your neural network, which is called brain, and will recognize most of the actors. So what we request from physicians is basically to recognize these actors just by looking at the nose and the lips. So this is not very dynamic and reliable. So, well, that has a history in medicine. And today we still use 19th or 20th century disease definitions and structures of medical records. ICD-10, by the way, is made for reimbursement, not for precision medicine. We name diseases by the organ still. We call them by the name of a doctor, Alzheimer's, that by chance started some ages ago or decades or centuries. to harvest symptoms, he found that they are lookalike syndromes, but most likely these syndromes are not really coherent and consistent and all the same. Even worse, we have just some kind of description of a status that we poorly know how to describe. This is my favorite. And that's exactly where we stand. In ICD-10, we have about 30,000 diseases. Unknown diseases just go in the drawer as dead people, but we basically do not really know how they have died. We know about 6,000 of these phenomenons, and we can treat about 3,000 diseases so-so. Diagnostics, not much better. Your chance to get a false diagnosis in your lifetime is about 100%. The good news is our body is quite robust and you might even survive the treatment of a physician. So the right treatment may take, the right treatment for rheumatoid arthritis, for example, is one of the major challenges that I am interested in. What about how precise are we with the treatment that we provide? You may know Humira, particularly when you're in the US, because you can't evade any kind of those spots that you see on TV all the time. So you have to treat at least 4 patients with rheumatoid arthritis. It's used for a plethora of other diseases as well, to find one patient that actually responds. So given the fact that this is the most successful marketed drug with about, I think, I forgot the number, but it's billions, you can imagine, that go down the drain because we do not know how many people respond to a drug before we give that to them. So then there is a new paradigm evolving. Actually, it's not that new. Clayton Christensen, who unfortunately passed away last year, he's an economist who was working at Harvard, described precision medicine as a paradigm shift in 2008 in San Francisco at a biotech meeting, where he was basically describing that today's medicine in the field is, and earlier on was symptom-based, intuition-based medicine. If you're lucky with your disease and you come to your physician, he's an old person that has seen a lot of patients and he may have the intuition what's really wrong with you. Or the same with a specialist who has seen a lot of patients and he might just look at you and get a feeling. However, we know this is really still very imprecise. So by chance, we came up with the idea that we are all the same somewhat, and we may just be placed into a cohort. And then we do these kind of clinical trials in cohorts, anticipating that all people are in the same state of aggregation, which is basically completely wrong, as I will show you later. And nowadays, with the advent of technology, high content data generation and computing capacities, we can actually use much more omics data, as we learned before, and aggregate them. And this is basically the foundation for precision medicine. We use all kinds of information that is available. So you have seen a similar slide before, so I'll keep it short. It's a little bit like generating Google Maps of disease, where you have all kinds of layers of information and you can zoom in, zoom out, get longitudinal snapshots, whatever you want. Unfortunately, these kind of Google Maps of disease do not exist at all. And in fact, it's not trivial to generate them because we are not like a car, static in a way. We are aging, we change our lifestyle, we basically change our diet, we change the environment. So this is all influencing the state of aggregation, which makes it a much more complicated, not even talking about ethnic background and what we do when we interfere with drugs in our metabolism and proteomic system, etc. So now I'm coming to John Ioannidis. Nowadays, he's actually very famous for being one of the heaviest critics of restrictions and non-targeted measures, if you want to follow his work. He's also one of the most cited scientists and respected WHO advisor. He came up with a concept early on that most clinical research is not useful, and that's just by the design of the study. Very often we see that study designs are not tailored to answer the question. I think most of you guys are engineers. I worked with engineers when I was in a consulting company where they were heading Mannesmann pilot development. And that's when I learned the major differences between engineering, physics, and other hardcore science disciplines and medicine, which is really very often intuitive and driven also by fashions, which we can also see in COVID right now. It's probably the only disease that sees that much redundant diagnostics worldwide for diagnostics purposes, but also for academic interest. And many of the data are generated in a very poor manner. So it's even worse. Up to 50% of what research which is done in the laboratory is not reproducible. And this has not improved over the last years significantly. Why is that? It's not always in purpose. In fact, I think this is rare. Mostly it's because there is some kind of irritation or what if change of protocol by a postdoc or a student, and you just can't find the source of this kind of change in the protocol which delivered a result that was very exciting, but at the end of the day, no one in the world can reproduce the data. This is one of the key problems when you get data. As a data scientist, don't trust the data. They might just be crappy. If they are not repeated redundantly, same again, COVID pandemic, people were pulling data from the internet, from various sources on whatever they wanted to prove from different countries, threw them together, not knowing which testing protocols were there, which reporting protocols. I know, for example, in some hospitals that had a high incidence, they were just not having a software that was able to differentiate between new cases and repeatedly tested cases. So this changes obviously the numbers that you generate dramatically. And this is the case in a lot of studies where simply the data cannot be reproduced in the same manner. So this is one of my favorite slides. Keith Yamamoto, he's the dean of UCSF, came up with the idea what we need, and this is crucial if we want to go in this direction. We need studies that generate standards, make all the efforts to make omics technologies comparable and reproducible. This is a key that is essential that we must establish, and we need flagship projects because we do not really know how the standards should look like. This is an ongoing process, and there needs to be a concerted effort worldwide to really avoid redundancy and resources and basically investments. So this is just a slide that demonstrates that the idea of generating longitudinal data, which are crucial, I will allude on that, to come up with the concept of precision medicine will be key to stratify patients better to the treatment or also come to prevention eventually. This is my hero. Sorry, I have a sore throat, not COVID. Mike Snyder is the chair and dean of Stanford Genomics Research Centers. Many of them might know him also for his NASA engagement with the twin studies. He did what some people call the narcissome. I think it's one of the key studies that were essential to actually show how in which direction we must go with precision medicine. He took 20 different snapshots over 14 months, generated 50 terabytes of data, and generated unique insight. For me, this study was mind-blowing because he showed, for example, that an asymptomatic infection, highlight, asymptomatic, completely screwed his complete state of aggregation. It changed his metabolomics profile, it changed his proteomics profile, but he didn't realize anything and he didn't feel anything, no symptoms. So what does this tell us? If we have a cohort where some people might just change the diet, might have a subclinical, formerly healthy state of asymptomatic infection, they might just screw up the data and we don't see anything. So the idea deriving from this study is basically that we should generate longitudinal multi-omics data and generate these maps of health and disease on a population scale level to really learn and understand more what factors influence our health and disease states, and then use an individual as his own control in clinical trials, whether he's changing from a positive, whether he's a positive or negative change, whatever kind of intervention, treatment, or drug we apply to him. So unfortunately, I have no audience, so I'm not sure if the point comes across. Usually I see eyes going up or they become critical. Well, there is a business case in that. And in the US, people try to generate these data. This is a QXM, a company by Andreessen Horowitz and Mike Snyder's team. They basically do this physical on steroids. You can have a basic membership, or the heavy membership, I think, is $20,000 a year, where they do this kind of multi-omics, MRT guided, super duper physical. And they do it over time. And this way, which is actually very smart, they generate this kind of Google Maps of disease. The problem, why do I mention this kind of a company? Not just to support it, but the problem is generating these data is not trivial and it's insanely expensive. So a couple of years ago, people asked, can we have a blood test that identifies approximately 20 cancer subtypes in the blood and would eventually also tell us in an early stage where the cancer might reside? People said, excuse me, people said this is basically impossible and insane. So how did they start this project? Grail, a company which is now being sold for quite some money to Illumina back, was a spin-off of Illumina and supported by Bill Gates and Jeff Bezos with a seed, and ARC Ventures with a seed funding of 100 million. Why was that money necessary? Well, they had a hint that with next generation sequencing, they could find some signatures of cancer in lung cancer patients. But they needed to prove that and also build a data reference database for other cancers. So they had a cohort that they were following over time. And they started in August, I think 2016, and they completed it just a couple of years ago. I cannot see my own slide, unfortunately, because something's hiding it. So Illumina, just also if you engage in these ventures, has generated a tremendous value. It was sold for 8 billion to Grail, and it's now one of the first tests. There are 2 or 3 other companies that have a similar approach, which is able to really identify free-floating DNA signatures of various cancers with a very highly precise way. And due to some kind of methylation of DNA, I'm not going into detail, we can actually figure out where the cancer might reside. These kind of technologies and approaches, although tremendously expensive, are complete game changers in the prevention and early treatment in oncology. Obviously, any cancer being treated the earliest we can will eventually go in full remission. So I think digitalization changes everything, and it's time to act. We need to build these flagship projects. May it be either industry, academic collaborations, may it be startups, may it be some kind of flagship projects, spin-offs from pharma companies. But what we should not do continue with the same thing that we've been doing all along, because this is not going to lead us anywhere. If we try to build the concept of precision medicine on yesterday's diagnostics, yesterday's taxonomy, we're totally going to fail. We need to build these Google Maps of health and disease. So what I'm also a little bit alluding on business model, insane ideas. Well, sell books on the internet, build a cheap computer for everybody, post CVs on the internet, and so on. So it would be a very good, maybe insane idea to reduce disease burden by precise health and disease management. So this is what Bill Gates is actually predicting. This will come in the future. And interestingly enough, companies that are leading the way, like Tempus, a company is started by a Groupon founder. iCarbonX, a Chinese company, is started by Tencent, and one of the founders of the former chief scientific officer of BGI, QBio, you saw before. Grail was started by people like Franz Och. He was the former senior data scientist at Google. He joined Foundation Medicine, and from there he went on to Grail. So I think data scientists can and should be the driver, although they have to educate the life sciences in academia and industry that we need more rigorously better, cleaner data. Otherwise, we only generate garbage in, garbage out. And well, coming to business models again, the one who has the data will run the show. And that's where I come to the end. I have a group on LinkedIn. You're cordially invited. This group is actually also searchable. So thank you again for your attention. I would love to see you all.
Thank you, Thomas, for your talk. Great as always. I like it. Yeah, let's see. Are there questions from our audience? Let's see one. So you can type in questions into the Q&A window. Until we get incoming questions, then let me ask you, Thomas, as you describe the status quo as not really satisfactory, what do you think would be needed most immediately from the constituents of the healthcare industry to change the status quo? Basically, how to enable truly precision medicine. What's the ultimate step one in your perspective?
Well, I'm tend to get a bloody nose with my comments. There is a great reluctance wherever you go to just simply do something completely new where we do not know the outcome. It's actually different in the pandemic. We do a lot of things where we have no president and no ethics committee and nothing, and we do experiments around the world. But when it comes to research in academia and industry, and also doing the clinical trials, people are most reluctant to do something new where they can be blamed if it's not working. One thing. Second, everybody shies away from the tremendous amount of money that is needed to generate these data, while the risk is relatively low, actually. When you look at the companies that were started with this kind of bold approaches, most of them were successful. Those that were not successful did simply not have enough money to generate enough data. And that's basically also true for many startups outside of medicine. If you have enough customer data, you can generate a successful business model. I mean, at the end of the day, it is a business. Medicine is a business, right? We have to appreciate that. So somehow we have to make sure that these data that we need are being generated, and the data sharing perspective is also one of the things that is a key in the future. I mean, physicians, I'm in Germany, go to a physician, ask him for his data. He would say it's his data. You're not having access to his data, right?
Fair enough. Yeah, and so I think that the data part is getting is getting quite important, as we have also seen in our very first talk, to have access to the real-time live longitudinal data. In the meantime,
we have the technologies.
Yeah,
we just don't have the data,
and we don't have the, I would say, international or let's say global same let's say policies and motivations. I think that's definitely something we hopefully.
Well, that's another thing. As long as a physician is being paid for the drug he applies and not for the drug that is successful, nothing's going to change.
Yep. Actually, in the meantime, we got a couple of interesting questions from our audience coming in. So the first one is from Satyajit Rao. So the question is, if you were to look at one thing that will change the future of health, other than precision medicine, what will it be?
But that's a tough one.
It's the IEEE. Welcome.
The one thing that would change everything, narcissism, eliminate narcissism.
So you would say mental health is the key.
In a way, I mean, I honestly do not know the answer. It's just our egos are very often one of the biggest roadblocks for getting innovations going, right?
Well, depends, but fair enough. Then let's move on to the next question from Memish Osan. Is the data collecting waste for data science in different studies cause ethical problems? Okay, interesting, yeah. Looking at HIPAA compliance, possibly.
Well, I mean, it's a major challenge because with these multi-omics data, you can basically not de-identify the data anymore. All those signatures are a signature which is like your fingerprint, like your face, whatever, you name it. And if you have enough information, there were longitudinal studies in the UK on metabolomics, and they could figure out what kind of drugs people were taking or what kind of underlying diseases they have. So the privacy issue, and we see that now, again, in the pandemic emerging, it's really a high risk of abuse, discrimination, whatever, if we have all these data at hand. So that's definitely an ethical challenge, yes.
Okay, very good. Thanks for this. And then last question from the audience. No, actually not last. There's one very interesting question from Nen Cho. Actually, I'm also a big fan of traditional Chinese medicine, so I like this question. So the question is, what do you think about Yin Yang, manifesto of the ancient Chinese Book of Change, describing environmental factors beyond scientific medical data, which may even go towards chronic inflammation as driven by lifestyle?
Well, believe it or not, I think we know, as you saw from my presentation, we know much less than we think about the health and disease universe. So I cannot exclude anything being as a rational approach unless I can disprove it. In fact, I'm a fan of Karl Popper because he came up with the concept that you have to disprove your hypothesis. And this is insanely difficult. And this would be a very interesting approach for lots of scientific concepts if we try to find the experiment. Just look at animal studies. You can find a plethora of parallels with mice. But if you come up with the experiment in mice that disproves that this is not, a drug is not going to work in the human being, that would be a game changer. But it's the mindset. We are stuck, and with Chinese medicine it's very interesting. They do a lot of metabolomic studies to find out why traditional Chinese medicine is working, because it's a mixture maybe between real drug effects or some psychosomatic. I originated from the Max Planck Institute for Psychiatry because I was interested in psychoneuroimmunology, until I realized that if I want to change the mindset to cure arthritis, that's probably beyond my lifetime. So maybe I should go to drug discovery.
Okay, fair enough. And so I think this is a quite complex topic, and we may not be able to conclude with the questions here.
But I love to engage in this kind of discussions. So people should invite me wherever they want.
Yes,
this is exactly my ambition.
Exactly. So let's take the last question from the audience, from Sergio Matos. So the question is, there are some communities trying to empower patients and people in general to take ownership of their data and share it for research. How far are we from such an achievement?
I think it's stupid, honestly, because they are not paying for the generation of the data. I think this data should be something like a public common good, be useful. And we see that now happening in Israel. I mean, Israel is somehow commercializing them, but they make sure that all people and individuals in Israel benefit from the data they are generating, right? And that's one of the reasons. So I think if there should be no bias, no group that is somehow opting out. I mean, if I go to the physician, I'm not paying for the blood. Well, I pay for it because I'm in a public health insurance system. But we should socialize the data. I mean, this is definitely not something that I'd like to see in a commercial pathway. I know that this is not mainstream thinking, and there's this discussion about data provenience and ownership. But I mean, if you benefit in a society from the healthcare system, you should be part of the data sharing effort.
Okay, very well. Thank you very much, Thomas, for this great talk and the great discussion. So I'm handing over now to my colleague Colin. He may have another question, but then I think he will start in the next talk thereafter. Hey, Colin.
Thank you. Well, yeah, you know, this is a topic that would, I agree, Thomas, would be a while. But we have
Thomas, Thomas, sorry to interrupt. Thomas, do you want to answer your screen? And then we can.
Yeah, how do I do that? Wait a second. I have to ask my daughter.
Okay, but don't leave, don't go. Stay here.
Yeah. I'll catch up with you later.
I have to unshare my.
That's great. So we'll move to the next speaker, and I'm very pleased to have
Thomas. We'll be in touch, Thomas.
Yes, we'll be in touch.
Great talk.
Thank you.
I'm very pleased to have Dr. Jadwiga Bienkowska. Is that? Please correct me if I'm wrong, because that's my pronunciation. I know you had told me a lot when we spoke. I want to make sure I pronounced it right. So am I close?
Yes, that was very good.
Okay, thank you. So Dr. Bienkowska is the Senior Director and Head of Computational Biology at Pfizer Oncology Research and Development, whose mission is the development of breakthrough therapies for unmet needs of cancer patients. Computational biology is used to decode complex molecular data into data-driven decisions to accelerate drug development, from new targets discovery and mechanism of action studies, translation of preclinical observation to patient selection, and progression of new therapies to phase 1 trials, and bedside to bench translation of insights from phase 3 to 4 clinical and biomarker data. Dr. Bienkowska has over 20 years of experience in pharma and biotech, assuming roles of increasing responsibilities and leadership to leverage data science and unravel molecular mechanisms of disease, advance new therapies, and deliver precision medicine to patients. Prior to joining Pfizer, she led computational biology research groups at Biogen IDEC and EMD Serono, focused on uncovering molecular drivers and new therapies for immune and neurological diseases and cancer. But Dr. Bienkowska holds a PhD from the University of Chicago and completed postdoctoral training at Harvard Medical School. So we're very pleased to have her here today. It's very special. And the title of her talk is Leverage Data Science and Unravel Molecular Mechanisms of Disease, Advance New Therapies, and Deliver Precision Medicine to Patients. So Dr. Bienkowska, please, the floor is yours.
Thank you, Colin, for a kind introduction. So I kind of generalize my talk to data science challenges in precision oncology. As you have heard from Colin, I am leading computational biology group in oncology research and development, and we work on the precision medicine, and in particular provide expertise in data science, how to manage and extract information from biomarkers. As you can see, biomarkers play a central role in precision oncology from the start. We use different resources, internal and external, in our target discovery, and then going on to mode of action studies to better understand how our potential targets work, and also use biomarkers extensively in disease models, and then later on in analysis of clinical trials and overall disease profiling. So we use the biomarkers and, as I say, precision oncology from the start, where in the process of target discovery, we are already faced with the decision of where, in which particular molecular context in specific cancer, particular target may work. And we use vast resources of data generated in public domains, such as CRISPR screens, functional genomic screens, single-cell RNA-seq, to better understand the exact context of the target, both from the respect of genomics, genetic alteration of the cancers, and also from the perspective of cell compositions of cancers themselves. This process and the utilization of the insights from the early preclinical work goes all the way to translations to the clinic. And in our later stage clinical trials, we again use very extensively molecular profiling to understand subtypes of the disease, and those learnings lead us to discovery of novel targets and kind of complete the cycle of drug discovery. The first one slide only that I have on more operational data science challenges that we face in industry is integration of different data types, in particular clinical and biomarkers data that are generated in our later stage clinical trials. We use extensively different CROs companies that generate biomarkers data and provide that to us, and then we need to do a certain level of QC to merge those with clinical data sets and generate what's called analysis-ready data sets. We often, through initial analysis of the first version of the data sets, we often discover problems with the data, even data generated by very well-established CROs, because we can, after merging of the data, identify the problems that are often lying in some basic data science data processing steps. So we have often the cycle of communicating back to CROs, telling them about our observations, and that leads them to refinement of their mostly analytical procedures to generate a better data set. So as you can see, there is a number of different QC processes that take place, and at this stage we are striving to make those processes as efficient as possible with various automation of QC checking reports, flagging out things that are inconsistent, etc. But we are not yet there. So in terms of the actual operational challenges for data science and practice, we need to be more automated in our QC processes. Let me just close this. I don't see my whole slide. Okay. So this is kind of enough about the operations. Let me go to the next slide. Okay, but I think the most important challenge in precision oncology and how data science can help to overcome it is tumor heterogeneity. As you all know, tumor is a very complex tissue that is made of many heterogeneous cell populations that cooperate to maintain the tumor. Those different cell types drive adaptation to treatment and lead to resistance mechanisms. And also, tumor itself is not a bunch of uniform tumor cells. It is made of many different clones, and the clonal composition of the tumor drives resistance as well. So we have been in the last few years very much focusing on trying to develop methods and understand better the tumor heterogeneity. How does that help us to understand the tumors themselves and leads to better therapy? In this process, we often are aiming to match our preclinical models where we discover the first biomarkers of target sensitivity of the resistance cells, patient-derived xenograft models and syngeneic models or GEN models for immune cancer therapies. We profile those models quite extensively on many different technologies that tell us how those different models represent the actual tumor tissues. And then when you consider the tumor tissues themselves, each one coming from a different patients, we typically look for the similarities across patient types and identify through those similarities biomarkers of sensitivity of resistance, leading us to more precise oncology therapies. So just to illustrate it on a concrete example, this is a transcriptomics view of tumor microenvironment heterogeneity. And this is data from recently published Javelin 101 renal trial that has been successful in application of avelumab therapy to treating cancer patients. With extensive molecular profilings of samples on this data trials, we have a much better picture of the heterogeneity. And what this heat map represents is on the top part, immune cell fractions estimates from the RNA sequence deconvolution, one of the methods that have gained popularity over the last few years. And at the bottom, there is a gene expression of specific genes that are associated with those different profiles. And that you can see from this picture, there is no two patients that are exactly alike in this representation of the tumor heterogeneity. However, there are clearly similarities across the patients. And we look how those similarities might be correlating with the response to treatment, which I will come back at the end of my talk. But first, questions that we often ask is how well our models represent human cancers. And in immuno-oncology, our tool for investigations of preclinical drug candidates are syngeneic models. We have invested also and extensively profiled the typical syngeneic models. And this two graphs shows the data from a paper published last year where we compared the syngeneic models versus cancers in TCGA. And the graph on the top shows the convolution into major immune cell types of the models, where we had two important high-level observation showing that syngeneic models tend to have much higher fractions of myeloid cells compared to human cancers. And also when you compare T cells fractions in those models to human cancer, they tend to have a lower fractions of human cancer and maybe not exactly representing the cancers as faithfully. Another observation of a difference between those models and human cancers in TCGA was a mesenchymal-like phenotype of syngeneic model as indicated on this graph by the round dots. And this phenotype is measured by ratio of fecal hearing to vimentin. It is an important phenotype since it is associated with resistance to multiple therapies. So overall, we think maybe the syngeneic models that we are using in IO therapies represent the more the harder to treat cancer types, which is not necessarily bad, because these are the cancer types that we are currently targeting to provide new therapies and options to patients. Another challenge in precision medicine, in oncology in particular, is continuous tumor evolution in response to environment, in response to treatment, and just natural tumor evolution. So the picture on the left shows a representation of the clonal fractions in the tumor that evolves in their time. Ideally, we would be able to collect serial tumor biopsies pre and post treatment, but for many cancers, those types of biopsies are very challenging or impossible to get. So recently, and you have heard from other speakers, a number of technologies that monitor cancer or response to treatments in periphery have been developed and enable to a certain extent to monitor tumor evolution and response. And those two technologies are circulating tumor DNA and monitoring of T-cell receptor repertoire in the peripheral blood. Even when you can have only get one biopsy at the beginning of the treatment, you can keep on monitoring patients through blood collections in as many time points as you wish, making it a very attractive way of looking at the tumor evolution and response to therapy. So in that space, we have invested quite a lot of efforts to understand and provide better and more effective methods of detection of circulating tumor DNA. The main challenge for detecting mutations in ctDNA is a very, very low number of fragmented DNA that comes from tumor that is circulating in the blood. So to improve the methods for detections, we have developed methods for improvement of this technology, and those have been published in a few years, and we are currently applying it in our own application of this technology. So here is a panel of genes that we have been monitoring in our breast cancer patients with digital, with ctDNA. I don't know what happened to my screen. Oops. Oh, okay, it's back. Sorry. And in this case, we have focused on the 87 genes and regions that are known to be associated and highly mutated in cancers. We have validated initially this technology with digital PCR and estimated the cutoff frequency for detection as 0.3%. We have since lowered it down to 0.1% and believe that at the level even of 0.01%, we have very accurate detection of the mutations. Okay, this is an example of results from applications of ctDNA to our clinical study of palbociclib and treatment of hormone-positive metastatic breast cancer patients with analysis of cell-free DNA collected pre and post-progression treatment. There are a couple of things that I want to highlight on this slide and representation of the genes that have been detected as mutated in this patient cohort. The first one is estrogen receptor gene, which you can see with the green bars is characterized by a higher frequency of mutations after the progression than before the progressions. And this observation indicates that partially the non-response to this combination therapy is due to the mutations in estrogen receptor itself and overcoming the fulvestrant treatment. Another interesting observation from the study was relatively high frequency of mutations, loss of function mutations in this case in RB1 gene. And this observation has been one of the important learnings that supports our follow-on molecules for treatment of patients that don't benefit from palbo, which is inhibition of CDK2/4/6 cell cycle genes. And we are currently conducting the phase 1 clinical trials with this new therapy in breast cancer patients. So a second technology that helps us move the needle in precision medicine is monitoring in periphery responses to the immuno-oncology therapies. The approved checkpoint inhibitors, anti-PD-L1, PD-1, and CTLA-4 have been shown to have an effect on repertoire of T cells in periphery. And on the right, you have a graph from the paper in Nature in 2014 for the first time that has been indicated the differences in T cell receptor repertoire at baseline, and the changes can differentiate between responders and non-responders. In this case, the protocol is to collect tumor biopsy and identify tumor infiltrating lymphocytes in the patient's tumor samples, and then monitor those tumor infiltrating lymphocytes in peripheral blood using TCR sequencing technologies. We have again invested quite an effort and collaborated over 3 years with Adaptive Biotechnologies, the provider or CRO of these technologies, and have developed a new method that allows us for a more accurate identification of the tumor-associated lymphocytes in peripheral blood. And on the graph on the left, you can see the comparison of the original binomial model applied to the data of PD-L1 blockade treatment in urothelial cancers. And you can see that with the improved model, we have a better distinction of the responders and non-responders as determined by percentage of tumor residence expanded clones in periphery. Again, in this type of technologies, the noise in the data is one of the main source of limited applicability. So any advancement that data sciences and new approaches can make in reducing this noise to extract the signal are very helpful. We are now routinely using the TCR sequencing in our later stage and earlier stage clinical trials to see, to monitor the response to oil therapy and identify subpopulations of patients that might be benefiting more and those that might need new therapies. So again, let me go back to the questions of heterogeneity and association of different biomarker styles with response. The main question that we ask in our analysis of clinical trials and trying to identify which patients' characteristics are more are beneficial or are associated with beneficial responses and which are associated with resistance. We again extensively analyze the molecular profiles of patients. Here is again the same picture from our renal cancer clinical trial, where it has been demonstrated that the combination of avelumab, which is checkpoint inhibitor, enaxitinib has a much better overall response than control treatment with the sunitinib. Both axitinib and sunitinib are VEGF inhibitors. The main learnings that we derive from such analysis is to identify a mechanism for resistance that lead us to new combination treatments and also new targets in research and discovery, while the mechanism of sensitivity typically point us to new indications that might benefit from this type of therapy. So in particular, in this trial, we have identified a predictive 26 gene signature that represents both adaptive and immune responses. And as you can see on the discovery cohort from the Javelin trial, 20 patients with RNA-seq data, and this Kaplan-Meier graph shows the avelumab and axitinib, where patients with the higher signature had a better overall response. This signature didn't differentiate patients in the control arm, although I'm not showing those graphs here. We have taken the signature in discovery from this trial and applied it to our earlier stage trial of only 49 patients treated with the same combination. And again, so the same association, basically showing that this predictive signature, which is now called Javelin 26, is really indicative of a good response to the therapy. Since then, we have applied the signature to other clinical trials of avelumab and still confirm the overall association of this gene signature with a better response to avelumab in other indications, in particular bladder, and that data has been already presented at ASCO last year, and it's now under review in publication. Another biomarker type that we have analyzed from this data is whole exome sequencing analysis of protein altering mutations in genes. Here again, we have identified a number of genes that differentiate between response to avelumab combination and control, and a number of genes that have been identified are associated with tumor progression and genesis genes that are kind of usual suspects, PTEN and mTOR, again validating some general knowledge about those genes, indicating that mutations in those genes typically represent more aggressive disease. Interestingly, we have also identified a number of genes that play a role in immune system control, such CD163 ligand and IL-16, and we are now pursuing those genes and observations in the immune space with our new immune therapies for cancer. So I'll end with acknowledgements. Of course, there are many, many people that contributed to the data that I have presented here. First of all, avelumab team and patients, and palbociclib team and patients, which are a big group of peoples. From computational biology, I have presented the data that a number of people in my group has been working on and publishing. And we have been very closely working with the biomarker lab on development of ctDNA technologies and panel, and we are continuously improving and expanding our panels and technology for applications in our early and late stage clinical trials. We have been working very closely with translational oncology leads for the clinical trials that I have been discussing with you, and our work wouldn't be possible without a great collaboration with them, and also our biostatistics colleagues are contributing to our development of new methods and then data science approaches by kind of keeping us honest on biostatistics and adaptive biotechnologies have been involved very heavily with us in developing better methods for TCR monitoring in peripheral blood in cancer. And with this, I'll thank you and look for questions.
Well, thank you very much, Jadwiga. That's wonderful talk. We appreciate your time. I think I could start with a question that if I understand correctly, that you've come up with a number of improved gene signatures that with existing clinical data, you've reinterpreted the data to show that in fact the progression or the PFS is extended over time. Is that right?
Yes.
Yeah, with that everything with the combination therapy. So what I'm wondering is that with that new insight, is that now going to be redeployed in the field to say, okay, you know, we can select patients based on this predictive 26 gene signature as to who should be on the combination therapy versus not? Is that now the next step that you're thinking of, or how will that play out?
I think at this stage we don't have such a strong differential in terms of this, in terms of this, the signal. I think the main learning from this is discovery of this signature and helping us in identification of other indications that might benefit, and that includes subtypes of indications like in breast cancer, maybe more post-progression, as well as looking at the opportunities of combination therapies, let's say between palbociclib and checkpoint inhibitor, where the question is timing, basically, when in the treatment course you should treat patients with one or the other, because we know that that often, at the progression time, both with palbociclib and after chemotherapies, typically tumors are not inflamed, and the general belief is that the more inflamed the tumor is, the more infiltrated, the higher likelihood of response. And from our studies of longitudinal data, for example, in chemo, we know that there is potentially a window of opportunity at about 3 or 4, 6 weeks after initiation of therapy. And we have some indication that it's also true in palbociclib case. There is a more inflamed tumor phenotype. So that's one of, I think, big questions in how to combine checkpoint inhibitor with other therapies in such a way that it will benefit patients.
Mm-hmm. Mm-hmm. I was also wondering, not to make your job any harder, of course, but Dr. Harris from the NIH talked about the different layers. There's different informational layers in biology, and of course, interaction between them and regulation going on between these different layers and such. So, would you anticipate including at some point in the future more information from on protein activity and things like that? Because right now it's really been focused on DNA mutations, manifestation, RNA translation, transcription. And so would you anticipate bringing this other piece of information at some point, and under what circumstances would you feel be appropriate?
So we actually haven't been talking about this data, but we do generate in this trial additional data, not on proteomics in cancer samples, because typically we have very little remaining for whole exome or for transcriptome sequencing, and use efficient, and sample saving technologies. Most of the samples are using clinical trials for approved diagnostics, such as PD-L1 IHC assays or CD8 assays that are directly linked to the goals of the clinical trial. So, we are lucky that we have some indications when there is more sample and goes to this, genomics technology. But given that, we actually do have data generated for peripheral blood proteomics. Again, those are not ideal because typically more noisy, etc. Nevertheless, we collaborate with a number of proteomics companies that generate data from periphery, from the tumor tissue. I think, as I say, it is a challenge because of the tumor sample limitations that we have. Typically, proteomics require quite a number of samples, amount of material, let's say.
Well, yet a grand challenge for the IEEE EMBS community. Great. Well, thank you very much. I really appreciate you coming and joining us today. That was a wonderful talk, and it's great work. And really, again, appreciate your time with us.
Thank you very much for inviting me, and I'm glad that I could share our research with the larger community.
So a pleasure. Thank you. So I'd like to move to our next speaker, Aaron Shellman. And Aaron is a technical leader and data scientist with a track record of building high output, delivery focused teams. She brings technical depth to all her work, whether that's informing company strategy, product development, or building and maturing analytical teams and organizations. Dr. Shellman is passionate about leading product-oriented teams to sustain rapid incremental deliveries. Effective communication is a core value of all our teams, and they continuously improve their data storytelling through presentations, technical writing, and peer editing. A common thread throughout her career is a focus on teaching and continuous improvement. She loves data science because it demands constant learning and evolution. I can see from your bio. So presently, Dr. Shellman is the head of data at Ginkgo Bioworks, and previously she held senior data science and analytics positions at Nordstrom, Zymergen, and Amazon Web Services. Dr. Shellman holds a PhD in bioinformatics and a master's in biostatistics from the University of Michigan, and a BA in economics and evolutionary biology from Case Western University. So we really appreciate her being with us today. And I'm really looking forward to hearing about her talk, which is entitled Data Science in Synthetic Biology. So Erin, thank you for being here with us today.
Thank you. Thanks for that introduction. And let me just get this shared here.
Great.
All right.
It works.
Cool. Cool. So I just want to start by thanking you, Colin, actually, for the invitation. And I'm really excited to be here and tell you a little bit about what we're doing over at Ginkgo Bioworks around manufacturing tomorrow in biofoundries. And so kind of like Colin said, I sort of started my career and my journey in data science at the University of Michigan when I was a bioinformatics PhD student. And I was working in the Brandt lab, which was a metabolomics core lab. And actually sort of working in data science before I knew that that was a career. So building lots of pipelines and doing data analysis on a broad set of research questions and working with a lot of different folks in addition to my own research. And then came out to Seattle and has been working in technology and sort of data science broadly, and have worked at a couple biofoundries, most recently as the head of data at Ginkgo. And so I'm super excited to tell you a little bit about what we're doing and then share with you an application in therapeutics. So biomanufacturing, I think, is really poised to revolutionize the way that we think about the creation of both sort of boutique chemicals, but also commodity compounds. And what we're trying to do in biomanufacturing is effectively apply the principles of manufacturing that we learned in other industries to engineer cells for various really super broad and sort of infinite number of applications that span agriculture, consumer goods, so things like fragrances and flavors, health and medicine, which we'll hear a little bit about today, waste remediation, and even plant-based foods. So if you've had an Impossible Burger, for example, you've eaten the outputs of a biomanufacturing process. And so where in other traditional settings, you might be at a refinery and see how chemicals and materials are made from that process. Here we're looking at a bioreactor where we use fermentation to make all kinds of molecules and compounds for applications all over the industry. And where if you were at General Motors, for example, you might see and hear talk about things like chassis that give us a framework for building vehicles of different types, sort of based on the same template and structure. We use the similar approach and similar language in biomanufacturing, except our chassis are organisms, and they're really sort of simplifications of things that you might see in a laboratory setting, or the model organisms that you use for a research setting can really be stripped down and characterized to create these templates that we then use to engineer. And so let's step back just for a second and explain a little bit about what we're doing when we're biomanufacturing or when we're engineering biology. So there's a lot of ways to do this, and this is just one that I think kind of explains what's going on. So effectively what we're doing is engineering microbes, in this case a bacterium or other types of cells actually, to induce it to do something that it probably wouldn't do in nature. Sometimes that's kind of overexpressing something it does already do in nature, or sometimes it's an example like this one, which is that we're actually going to insert a foreign piece of DNA, in this case, a human insulin gene, onto a plasmid, and we're going to insert that into the bacteria to induce it to express that gene and produce insulin in its environment. In this case, we'll stick it in a fermentation tank. It's going to eat it's going to consume energy, and it's going to excrete insulin into the environment that we can then purify those extracts and then have insulin or therapeutic ready for distribution. So again, lots of ways to do this, lots of organisms to work in or cells to work in, lots of ways of engineering them. But ultimately it's a process that will look similar to this in the application space. So where are we doing this work? Well, we're doing it in things called biofoundries. And biofoundries really are a very cool place to work because they integrate lots of different cutting edge technology from different disciplines. And so we're working at the intersection of really cutting edge molecular technology and the ability to synthesize and assemble DNA that we design, robotic automation, software and hardware systems, and then data science. And the goal here is to really standardize and automate big pieces of our design, build, test, and learn cycle, or the DBTL cycle, which you may have heard as sort of just a workhorse way of thinking about progressing and iteratively improving. But it's really core to what we're doing in biofoundries. And again, what we want to do is apply the lessons that we've learned from traditional manufacturing settings to improve our ability to engineer microbes at a high quality and to do that fast and to do rapid prototyping. So where there's a lot of commonalities with sort of what you would do in a laboratory setting or at a university doing research, but our focus is often really different in this space. And so we're very, very concerned around things like process optimization, actually thinking about the way that we work and what is effective and not effective. High throughput screening, so being able to do things in parallel and test a lot of hypotheses simultaneously. Like I said, with chassis, characterization of composable and reusable parts, so making it easier for us to engineer biology. And then rapid prototyping. So some use cases, some examples and applications are easier than others. And so we want to accelerate that prototyping phase as quickly as possible so that we can fail as soon as possible and pivot our strategy if we find that something is going to be a challenging application space. So all of this really is in service of making biology easier to engineer, and that really is has been and will always be the mission of Ginkgo. So the DBTL, the design, build, test, learn cycle, is really the heart of our foundry at Ginkgo and foundries in general. So what I wanted to do today was walk you through the steps of this process and sort of talk in general from a data science perspective about what types of questions we're thinking about at each of these steps, and then what types of tools and methods that we would use to answer those questions at each of these steps in general, but then also anchor that to an application and sort of a collaboration that involves health and therapeutics. and show you a cool application of the use of our platform for optimizing a therapeutic. So this is work that was done as a collaboration between Synlogic, is another synthetic biology company, and us at Ginkgo. And this is really all about optimizing a strain, optimizing a therapeutic idea so that it's more efficacious for patients. So the case study is designing therapeutics for maple syrup urine disease. So this is a metabolic disorder, relatively rare metabolic disorder, and it's caused by a defect in a multi-enzyme complex that ultimately metabolizes branch-chain amino acids. And so the result is that people with maple syrup urine disease can't metabolize those amino acids, and so they build up to toxic levels in their circulating blood and in their urine, and that causes sort of a sweet-smelling urine, sort of like maple syrup, which is where the name of this disease comes from. It's not a fun disease to have. It creates a lot of organ toxicity and sort of neurodegenerative issues. And so having great therapeutics here would definitely be an improvement. The treatment landscape is a little bit polarizing, so there's not a lot of options. On one hand, you've got dietary restriction and monitoring for life, where on the other hand, the only curative option is a liver transplantation, which has limitations for obvious reasons. And so the problem statement here really is there an opportunity to create a therapeutic sort of in between that would help improve quality of life for people with this metabolic disorder? And Synlogic had reason to think yes, based on past research where they injected C13 labeled leucine into mice and observed that a lot of that ends up in the small intestine, which lends credibility to the idea that we would be able to target therapeutics to the gut or to the small intestines, sort of the same way that you would take a daily probiotic by introducing synthetic microbes to that space to be able to metabolize those branch-chain amino acids and actually reduce the circulating amount to ideally non-toxic levels. So Synlogic came to us with a prototype organism that did that, and a prototype pathway, degradation pathway for leucine. And the question for us at Ginkgo is, could you take this prototype that doesn't quite meet a therapeutic bar and optimize this to get better performance and better degradation of those branch-chain amino acids? So here's what that looked like. So there's a sort of a 3-enzyme pathway here. And we'll focus, just for the sake of simplicity and just walking you through mostly the platform, focus on this first one, the leucine dehydrogenase. But effectively what we're doing here is starting with one of these amino acids that we don't want to accumulate and breaking it down through a couple steps with these enzymes into a byproduct that we're not as concerned about. And so the goal here is to take these 3 enzymes and really to optimize them to be more efficacious and to have characteristics that get us better outcomes for ultimately for patients. All right, so now we've got our problem statement. We have 3 enzymes that we want to optimize, and now we're going to drop that into our platform here. So the first step of our process in the DBTL, design, build, test, learn cycle, is design. And so this is a really, really cool place to work, especially as a data scientist. And the reason is that this is both the beginning and also the end sort of your loop. And so this is all about idea generation. And at its heart, what you're trying to do is produce a bunch of ideas that you're going to ultimately screen and kind of experiment on or test with experiments. But the sources of those ideas are really variable. And that's where you can really flex on some of these advanced methods, advanced analytical methods in deep learning or in machine learning and AI to generate new ideas, new candidates of things that we're going to build and then test. That's one approach. Another approach is that you can mine for that biological diversity from metagenomic libraries. And that's what we did in this particular case. But ultimately, conceptually, what we're doing at the design phase is generating ideas that might get us the sort of the outcomes from the engineering perspective that we're looking for. And then we're also designing conceptually the sequences that we're going to build in the next step. And so here's what that looked like for us. So in our case, Ginkgo has something that we call the code base, and that's a huge library of biological variation and sort of reusable parts that we can go to as anytime we have new problems to ask, are there solutions from biology that we can apply in this space? And so in this case, what we did was go to that library and find the same enzyme represented across different types of places like bacteria, fungi, animals, and plants, and find those similar enzymes. And what you're looking at here for this leucine dehydrogenase is the red dot. Oh, sorry. The red dot here is showing the prototype strain and sort of where that enzyme sits. And then what we're looking at is the enzyme similarity between these other candidates from different organisms. And so what you can see, these are all, effectively, they're doing the same thing, but their sequence is different and they have slightly different, probably slightly different characteristics. And so our question is, are any of these good ideas? Do any of these have the characteristics that we're trying to find in the activity of this particular enzyme? So we started at the top of our sort of funnel here with about 1,400 enzymes identified or designed. We screened most of them, and then we ended up with about 220 enzymes that did pass that preliminary screen and go into a second round of screening. So ideas generation. So that's design. So that's sort of the first step. And what's important there is that at this point, everything is conceptual. So again, ideas generation, what is my strategy for finding things that are going to give me the engineering outcomes that I want? But I haven't done anything yet. And the build step is really sort of where the rubber hits the road. So this is where you actually execute on those ideas, either coming from algorithms or coming from biological diversity. And we actually build the DNA. We build and sort of go through the assembly strategies. We're doing a lot of sequence validation and alignment type of work. And then of course, the transformation of those cells themselves, actually introducing that new synthetic DNA into the organism and then validating that it did, that it integrated the way that you thought it did, and it's doing what you think it's doing. So where design is sort of, in some ways, this big landscape that we're going to crawl around and explore, build is very operational from a data perspective. This turns into the types of things that you're going to be thinking about here are more around quality control, making sure, again, sort of at lots of different granularities, making sure that we actually were successful in building the thing that we designed. Lots of tracking, lots of liquid transfers. There's a lot of operational considerations here. And then another role that a data scientist might play, collaborating with the build group, is potentially helping to characterize reusable parts. And that's sort of what we're looking at here as an example of, this is actually a chassis strain for yeast and what we're seeing are sort of them developing puzzle pieces that we can reuse, recomposable blocks to accelerate engineering applications in yeast in the future. And so what a data scientist might do is partner to help characterize the performance of these pieces across different contexts so that we understand when we would pull this off the shelf to use it versus when we wouldn't. So very, very kind of a very cool space. This is where there's also just been a lot of, I think, innovation and sort of liquid transfer and handling of the amount of volume and samples that we have in this space. So coming out of build, so we've designed our ideas, we've built our ideas now, the ones that are buildable in strains. Now what we've got are strains and they're ready to be tested. So this is when, again, sort of an operational view, but lots of really fun data questions then at this point. So our focus is really defining scalable assays and then screens. So assay development is a huge challenge and an interesting problem in this space, which is really, we're going to ask we're going to test a lot of hypotheses in parallel. So how do I take how do I basically scale my experiment so that I can fit it all on a highly dense plate and ask a bunch of questions all at the same time? And then how do I do that in a way that actually correlates to performance at scale too, in these big fermentation tanks where the product in a production setting will actually be made. And so there's a lot of things to consider at the level of assay development, and lots of statistical kind of parameter search questions there. High throughput screening, if you're like me and have a background in stats, that's a very interesting place to work, because that's where, you know, all of these sort of traditional statistical questions really come into play. So you're thinking a lot about design of experiments. Do I have enough power in my test to even detect differences? And sometimes those will be small differences. Does my replication strategy support my screening strategy? And we'll talk about that in a second. Do I have a clear decision criteria for how a candidate strain gets promoted from one round of screening into another? And really laying out sort of, again, that analysis plan and sort of an operational plan for how we interpret the data coming out of the test process. And then last, but definitely not least, but often I think treated as least, is stopping and thinking about where you are. You're in the test step, which means that the next thing you're gonna do is the synthesis step, where we're gonna learn, and then from what we learn, design a new round. And so it's important to think about what's going to happen next, and to structure your data and your outputs at the test stage to support what's going to happen next. And so humans, if the other end of this test cycle is a scientist, humans are much more flexible and are able to look at variable outputs and to understand what they're looking at, synthesize information, and then move on to the next step. If you've got an algorithmic piece of your cycle where data is going to flow in and an algorithm is actually going to help you design what you're going to do in the next round of screening, you have to be a lot more structured about what your data looks like, because machines have much higher expectations and sort of requirements for what that looks like. And so even at this stage, you really, earlier, you want to think about what the data is going to look like coming out of this, and make it amenable for algorithmic consumption. So thinking again, sort of as a practical example of the types of things that we're working on in the test step, the replication strategy that supports our screening goals is an interesting part and an important part. And so generally speaking, you're going to do multiple rounds of screening, and it's again sort of like a funnel. This is funnels inside of funnels, where you're going to start with a lot of ideas and you're going to distill those down just to the best ideas. So at the first level of screening, your goal ultimately is to minimize false negatives. So you're actually going to be more permissive. I'm willing to accept more error at this phase as long as I don't lose the best ideas. And so operationally, you're talking about many more hypotheses or strains that I'm going to test, relatively low sample sizes, relatively low replication, and then a low promotion threshold. It's not that hard to get promoted into the next round of screening. So to give you an example from the SynLogic work, the initial replication strategy was just 2, and then we take the maximum, and that represents the performance for that strain in the first round. And then in the second round of screening, we're doing sort of the opposite. We want to minimize false positives, because the next thing we're going to do is expensive. So at this point, we want to really distill what we've got and make sure that what we're looking at are actually better performing strains than what we're benchmarking against. So fewer hypotheses, fewer strains at this point, bigger N, larger number of replications, and maybe more complicated types of replication, and a higher promotion threshold. So at this point, we might actually formalize this with a statistical hypothesis test and use a p-value threshold to determine what goes into, in this case, our tank validation, which is an expensive step. And so we're going from large to small, and our replication strategy sort of supports that. And it works. So here's the result from the leucine dehydrogenase for that initial enzyme in our 3 enzyme chain here. And what we see here is where we started, the initial prototype strain. And what we can see is that we've got a lot of candidates that are outperforming that, and in some cases outperforming it by almost 2-fold. And so great results from a primary, or sort of the initial screen. Another thing that we can do here is sort of filter based on other requirements. So our main requirement is that it outperforms our benchmark prototype, but another requirement might be that it's specific. So those branched chain amino acids are all sort of structurally similar. And so that enzyme can actually work on metabolizing isoleucine and valine also, but we don't want it to. We want it to only to be specific for leucine. And so another filtering criteria at this point in the screen might be for specificity as well, if that's something that we can measure directly, which in this case we could. All right, so now we've designed our things, we've built them and engineered the microbes, and then we've tested them and we have results. So now we're at the learn stage, and this is again another sort of a really fun, they're all fun, but a fun place to work because it's just there's lots of different types of directions you could go and lots of problems to solve. So the first thing you might want to do at this phase is consolidate your best hits as candidates for the second round. So I might take my best enzymes from each of those three and put them together with the hope that the best three are actually, I get more than those additive effects when they're together. I might, if an algorithmic cycle was part of your workflow, this is where I would update the models to reflect all of the data that you just generated in the test step, that test screen. This is where we could also prioritize process improvement. And that's actually stepping back and asking about how we're working. And are there parts of the process that were bottlenecks, or are there parts that injected a lot of process variability into what we were doing that we could eliminate or improve? This is also where you might collect more information from other types of experiments. So you might run your sample through some metabolomics or do some validation runs in the fermenters and integrate that information here to help design your next round. And so that's what we did using those pathways. So we were able, as you saw, we were able to identify improved enzymes, multiple improved enzymes over the prototype. And so the next step is really to take all of our winners from each of those three, and we've been talking about the first, but each of those three enzymes, and to stack them, sort of combine them and ask the question, what is the right optimization of the combinations of the enzymes that we found to optimize the pathway, not just the individual enzymes, knowing that your best three might not, when combined, to be the best ultimate performance. And so you want to search that space. And so here's our design outcome effectively for a second round of experimentation, or second churn of that wheel. And again, able to find through that other combinations or optimized combinations of those three enzymes that got us better results than our baseline. And then the last piece, sort of the validation piece of this, is this study using non-human primates and trying it out effectively, trying it in a more realistic setting to see, do our screening results actually correlate with the thing in practice that we want to do? And so challenged a bunch of monkeys with a high protein meal or slurry, I think. And then they got one of sort of two controls, either the base strain or this vehicle, or the optimized strain. And what we see is that the optimized strain does perform better than where we started in terms of metabolizing and clearing circulating leucine from these monkeys. And so the platform was a successful application of the platform to optimize this particular therapeutic. And then Synlogic did a couple additional screening and optimizations based on the designs that we produced. So again, sort of exploring the host strain. So could you use would you get different performance for different microbes? The bioprocess optimization, again, sort of our workflow, is that supporting our goals, or are there places to improve? Promoter testing, which is almost more granular optimization. So actually being able to have more granular control on the expression of those enzymes. And then of course, this validation of actually applying the optimized strains to a more realistic setting. So with that, I'll leave you with I love this paper. If any of this was interesting to you, I highly recommend this paper. It's a fairly recent one, and it again goes through sort of this example of the DBTL cycle and how specifically how machine learning can be used to accelerate our progress through that cycle. But I think that they do a phenomenal job of just summarizing, I think, the biggest challenge for data science and for biology in the biofoundry space. which is really to make the point that while the potential is large, the impact on the sort of the chemical industry broadly has been somewhat muted. And that's, as they say, historically because strain development has not been a wholly systematic enterprise. Instead, genes and their expression levels in the chassis organisms themselves have often been tested in a trial and error basis, typically informed by biochemical intuition, in order to eventually settle on a strain with production metrics and suitable scale up. And that process isn't actually that much faster or less labor intensive than traditional ways of getting there. And so I think the biggest challenge for us in this space is really learning the lessons that we need to learn from manufacturing, and then understanding and sort of characterizing biology in a richer way so that we understand where they apply and where they don't. And we can really strike that balance. And so with that, I just want to close by saying that we're hiring. So if any of this was interesting to you, please reach out, and I'm happy to tell you more about where we're going and what we're doing, and would love to talk more.
That's great. Thank you very much, Aaron. That was a wonderful talk. Very interesting. I think we have time just for a couple of questions. You know, one that came to mind is that we sat through a whole bunch of talks earlier about cancer and how cancer comes about through variation in the genome as a result of division. And so I was kind of curious in this case, you know, bacteria, you know, they're very, they reproduce themselves very quickly. And I would think that that could be a source of variability relative to the kind of modules you're putting together and building in a chassis. But then how do you deal with that variability in your algorithms and the predictive models that and the design models that you've been working with? Is that an issue at all, or is that just something that you build into the overall design, build, and test process?
Yeah, I mean, I think you're hitting on something that is also, I think, a big challenge, if I'm understanding your question right, which is how do we kind of leverage the variability? In our case, we want that. Like in the last example we saw is in a way, the premise is that biology has probably solved this problem for us already. And we just need to find the one that fits the solution we're looking for. And so I think there's a number of sort of challenges and interesting places to work in that space. And one is that just being able to mine the biological variability and to identify similar pieces and to extract them. And then the other is to characterize them and describe what they're doing, what their role is, and how they perform. So characterize both in sort of the annotation and almost the bioinformatic view or biological context on what is this piece, and then characterize also experimentally to say, well, how does this piece perform? And the context piece is actually a really hard thing. And that's something we think about a lot in terms of how do we represent scientific intent, or how do we describe our strategies for thinking, for navigating this really big biology kind of space, and these strategies that we can apply to engineer it. And that's unsolved.
Yeah, and the modules you build you string together into a chassis, are they linear or nonlinear? Because I would imagine, you know, linear models obviously are more, you know, easier to predict the outcome, but a nonlinear model or nonlinear elements tend to give you greater diversity potentially, but are much harder to predict what's going to happen. So
yeah, I think it really varies, and it varies a lot by what you're doing and kind of what type of information you have at what time. And so to give you a sense of the sort of modeling variability, I think of something like flux balance analysis a lot, which is a very, you know, sort of your workhorse methodology for metabolic engineering. You can do that without any data. And so that's any actual experimental data. So that's a way for you to kind of crawl around and explore computationally versus, but that's sort of a constraint-based modeling technique versus sort of the kinetic version of that, where you've got all these parameters and you're actually describing the change of metabolites over time versus sort of these steady state things. What's really neat is that there's increasingly a machine learning approaches to this that actually blend both of those perspectives. And so you can kind of create both the dynamical outputs with more of the simplicity of a constraint-based approach. But what's neat about it is that in some ways, from a more, like you can almost ignore some of the more statistical limitations or the subtleties of what the models are doing, because ultimately these are ideas coming into an experimental platform that we're going to vet. And so it's a neat space because we don't need to be that precise in a way. We need to have a process that generates good ideas, and they should be better, you know, the more and more we do this to help narrow our scope quickly so that we can move faster. But we just need to have a direction. So we're not inferring things from the model. We're not always interpreting models. We're just using them to generate new things to engineer and test. And so it gives you, it's more of an engineering application than it is a statistical one in a way. And so it gives you a little bit more flex to try things or to maybe apply models in spaces where they're hard to understand or explain, but what you're explaining is actually how they perform versus the models themselves. I'm not sure if that's what you...
Well, that's great. That's fantastic, Tom. Really, thank you very much for joining us today. That's really awesome. I'm really glad to put this just jobs. You're hiring. That's great. Wonderful news. So now I'll hand it to Ali, who will introduce our final speaker for today's session.
Thanks again, Evan. Hey, I'd like to introduce now my former colleague, Anita Gogat, who is with HP. She's leading the tech scientific incubation in the HP labs and CTO office. And she will be talking about a very exciting topic, because at the end of the day, we talk about all these fancy, let's say, approaches of, let's say, monitoring and testing. But at the end of the day, we need to have the technical tools based on microfluidics, MEMS, and nanotechnology. And that's what Anita will be talking about. Hey, Anita, great to have you here.
Thank you so much for the introduction, Ellen. It's good to see you again. I was going to actually preempt the talk saying it's going to be a little bit different from the prior conversations, because it's really going to focus on more of the engineering aspects of the engineering tools that are needed to actually not just generate this data, but I think this talk will ultimately give a little bit more perspective on what kind of technology convergence and tools we need to deploy in order to now leverage the information and insights that we've acquired. And now we could actually deploy these more customized, more precision, in vitro diagnostics or therapeutics tools. So again, so this is a talk on more engineering focused perspective in generally healthcare and precision medicine. I bet most of you have come across HP at one point. We are a pretty well recognized brand in the compute and print business, but not a lot of people know who we are under the hood, if you will. So in this talk, I will give a very brief introduction to the HP innovation history, as well as a sneak peek under that hood, and provide some insights, market insights about opportunities for microfluidics, MEMS, and nanotechnology. So Dave Packard and Bill Hewlett founded the company in 1939, and landmark inventions included the audio oscillator, the world's first desktop computer, or the world's first scientific calculator, handheld calculator, the first printer, and also the first generation of lab-on-a-chip technology that integrated chemical manipulations in a single chip. At this point in time, the measurement systems, as well as the life sciences technology, was spun out and became Agilent, which is a really well-recognized entity in this industry. And in the next 20 years, I think it's safe to say that these initial inventions have shaped many industries, including compute, print, data management, measurement systems, and life sciences. In 2016, we've looked at manufacturing practices today and then actually found massive opportunities in customization through 3D print. So we have actually launched our own 3D print business and leveraged our multi-jet fusion to reduce cost, introduce more speed, as well as give new material opportunities in 3D printing. And then most recently, we started revisiting another significant industry, which we see a lot of interesting trends in, which is healthcare and life sciences in general. So the healthcare spending is on the rise, as we all know, driven by aging and growing population. But beyond these global economic factors, we wanted to look at it more closely. So we saw some mega trends pointing to a transformation about to take place within life sciences. And many of them were mentioned today, and I'm just going to highlight a couple of them here. One is, especially now within the pandemic era that we're living in, we're seeing now the acceleration of that demand for decentralized healthcare, as well as, thankfully also the supply for decentralized healthcare and solutions for them is also increasing and accelerated. We're seeing also a change in reimbursement strategy from fee for service to fee for value. And then finally, also the emergence and the growth of drug-resistant superbugs, and now this globally ubiquitous, highly contagious viral infection caused by this new strain of coronavirus. So these are pretty big events, pretty big trends that are happening and have major technology implications. So some of the ones that we've kind of started to internalize include looking at many of the solutions that are addressing the needs are fairly complex, involve very expensive reagents, very precious resources. So miniaturization and automation as an implication is a significant implication for this area. The other trend or implications that actually this entire conference is focusing on, how do we merge these novel and powerful technologies to deliver more precise and more accurate insights that improve patient outcome. And ultimately, the last one I would kind of highlight is there's an enormous pressure to reduce the cost of these solutions as well as improve access. So these all together kind of evolve into a fairly large pressure on this industry to deliver great value. And then we took it a little bit deeper because in a few minutes, I'll show you what HP fundamental technology is. But we wanted to understand how HP's foundational technologies can actually ultimately add value in different segments within life sciences. So we broke it down to in vitro diagnostics and therapeutics. And within in vitro diagnostics, we saw there is a significant driver that's associated with lowering the cost of the solutions for these diagnostic systems, as well as improving access. And these would include applications like the typical blood panel and drug screen for triage, infectious disease, which is very on the front of everyone's mind today. And we've talked, and there was several talks discussing companion diagnostics today, companion diagnostics that would guide therapeutic approaches for the patients. Now, there are a lot of different approaches to try to lowering cost and improving access. One major one, and as that is pursued by a large numbers of microfluidics companies, is to try to integrate a lot of the complex workflows on a consumable, a highly integrated automated consumable. And it's a very powerful approach, but our own primary research has suggested there are some remaining challenges here for the clinics and hospitals that are adopting them. When these workflows are adopted or implemented onto a consumable, the entire design is fixed. And often it takes a few years to introduce a new test on it. So when a clinic invests into an instrument and a test, they have to train their staff on it. And if the test menu is not sufficient and they have to wait a few years to introduce a new one on that system, they have to invest into multiple capital equipment, they have to do training, they have to buy different consumables, and that's a lot of pain point for them. Now, it's also this full integration onto the chip for things that needs to be a little bit more customizable or we anticipate a lot more faster introductions of new assays and targets, it is a constraint. So when we looked at this space, we really wanted to understand how do we bring the benefits of miniaturization and microfluidics and these nanotechnology tools to this space without over-constraining necessarily to a fixed design. So that is kind of a driver here that we're kind of trying to digest and act on. And then the second subcategory within this space is enabling those new advanced functionalities that bring greater precision in the therapeutics that is guided by this diagnostic answer. A couple of examples here. One would be, for example, antibiotic susceptibility testing. It's not enough that we diagnose a patient that they have an infectious disease or even what pathogen is causing the disease, but we really need to understand what is the right drug therapeutic approach to address that infection. And the best way to do that is to isolate the pathogen and then test the drugs and different cocktails of that drugs and measure the effectiveness against those pathogens. So and because of that, because of the pathogens often mutate, as well as new drugs are introduced into the market, a test system that can retain some flexibility to introduce these new additions or modifications is critical for this space. The second area is in the therapeutic space, and I'm just highlighting here this traditional pipeline for drug delivery, which is very complex, includes discovery, formulations, validations, quality control. All of them involve complex workflows, expensive reagents, and often use very precious animal and human specimens. All of these making this pipeline into a prime target for improving with microfluidics. So again, as long as we retain some of the flexibility in areas where there's a lot of change from one workflow to another. But then the fourth category and the second one within this therapeutic space is moving into a more customized therapeutics. So whether it's a customized prescription drug that we would print for a patient in a drugstore, or it's a personalized gene therapeutics, we understand that the traditional analog manufacturing methods that were designed for high volume manufacturing and the standard formulation, they're not going to be meeting the requirements for this space. A more digital, a more customizable approaches are needed here to address this space. So this is kind of what we saw at a high level in areas of opportunities for us. And I'm going to try to share a video about us. And I'm going to change briefly a setting so I could actually improve the quality. And Ali, if you can just, if I can ask you, let me know if there's no sound, because in that case, I'm just gonna stop the video and I'll just share some snapshots.
Yep, just go ahead. Audio is coming.
Thermal inkjet nozzle chamber is thinner than a human hair. Yet it's the fundamental building block of our print business. Tiny resistors superheat the ink to 340 degrees Celsius in a microsecond. 6 million resistors cover 1 square inch of our print heads. This innovation powers our leadership in new areas like graphics and 3D printing, and makes us the world's leading microfluidics company. We double our thermal inkjet performance every 18 to 24 months, and can now jet more than 25 billion drops per second. It is as revolutionary for liquids as the microprocessor was for electronics. These microfluidics processes can now control, mix, sense, and analyze fluids as small as a single blood cell, blending and routing multiple liquids at a microscopic scale. Imagine hyper-accelerating the testing of new antibiotics that can fight drug-resistant bacteria. Imagine instantly dispensing complex medicines whenever and wherever you are, opening up a world of personalized digital healthcare. That's disruption and reinvention of markets.
I'm happy to hear that the audio came through. So hopefully I changed back the settings. So this brief video was a very high level introduction to what the mode of microfluidics that we've been using for our core print businesses, which includes 2D print as well as 3D print. And then, as I mentioned earlier in the introductions, we are now trying to repivot some of these assets and see what kind of pain points and what kind of value we can add into the life sciences space. And in the next 10 minutes or so, I'm going to try to give a few examples of what we've been reducing to practice and how do we really use these assets. But there's a lot of different directions we could take this. And I'm hoping just with this portfolio of examples, you'll see kind of the opportunities that we may be able to address together. So the first thing that was really a low-hanging fruit for us is how do we introduce the benefits of this precision in micro scale fluid manipulation without integrating it into the chip. And the approach was, we're going to bring the microfluidics advantages to the plate-based reactions. We can jet at the 2 picoliter volume resolution and worked with several laboratories in a few applications. And here I'm showing 2. In the first scenario was antibiotic susceptibility testing. As we all know, a patient comes into the hospital with an infection, they put on an antibiotic, and if they don't respond to any of the standard antibiotic courses, then the hospital has no choice but to send the patient specimen to the CDC, which is the only agency that has the authority to test unique combinations of antibiotics. And even if there was an antibiotic that was recently approved by the FDA, those usually don't show up onto these more automated panels and provided access to the hospital. So there is typically up to 3-year gap before that new drug is available. So the CDC then uses a standard approach, gold standard approach to do this antibiotic testing, but that introduces about 7 to 10 day delay for the appropriate guidance for what antibiotic cocktail would be effective. And for a patient that has severe infection, has sepsis or even septic shock, that type of a delay can be catastrophic. So there's a strong need to bring that type of complex combinatorial testing nearer to the patient. So the CDC worked with us, and we gave the CDC access to our dispense technology to see if we can improve we can simplify the complexity of this setup of the combinatorial testing, if we can improve the accuracy and precision by this approach. And after thorough testing at multiple centers, the CDC made the recommendation to make this type of dispense platform a gold reference method for preparing combinatorial reactions for antibiotic susceptibility testing. This means that any hospital or clinic in the US could use this system to perform this more complex EST testing. And the second category is we've worked with labs that have used this dispense technology again on the plate and leverage the digital and micro scale aspects of the technology to, one, reduce the amount of expensive resources that are used for these plates, reduce the amount of specimens, animal or human specimens that are used for testing, as well as improving the precision and accuracy also by the fact that we have very precise dispense, but also can implement very complex plate layouts, which with a robotic system that are typically used with plate-based reactions is not possible. And in this case, OSU and also Merck have implemented BCR and ELISA workflows and found greater performance, the elimination of plate bias, as well as an order of magnitude decrease in precious resources for these assays. So that was kind of a lower hanging fruit for us. And then we started thinking about how do we now bring in other and converge with other interesting technologies. And the first thing that we did, and here I show a SEM image of a sensor or substrate called surface enhanced Raman spectroscopy substrate, which is a substrate that will amplify the molecular signature of a molecule that binds to it. So it's not only able to detect it, but it will be able to identify it. And these substrates, their function is to amplify that vibrational signature substantially. And the one that we have produced in our laboratory can amplify the signature by at least 10 orders of magnitude, if not higher. So we have single molecule sensitivity of these sensors, and we've deployed these to detect contaminants and economic adulterants in infant formula for a couple of years, and we worked with the FDA on this. And then we shifted to try to implement them for rapid AST testing by measuring the volatiles coming out of the bacteria during culturing. But actually what we found is that while SIRS technology is actually very powerful technology, what it couldn't do is to perform the measurement reproducibly and enable quantitative measurements. And that's a major pain point within this technology space. So our team has worked on trying to come up with a way that can calibrate these sensors, and we can make highly quantitative measurements. So here I'm showing an approach where we converged a microfluidic method, the dispense method, with a powerful external sensor, and we supercharged them by coming up with a single chip calibration method, where we can make 200 independent measurements on this single 1 millimeter scale sensor with a quantitation resolution of 3% or less. And this for us was a very big breakthrough, because in this industry, it's very difficult to achieve this kind of reproducibility. And the good news is that we're completely agnostic to the sensor. This convergence doesn't have to happen with our microfluidics and our sensors. It could be any other SIRS substrates or any other type of modality of sensors as well. Another category of convergence that we have been pursuing with passion is on-chip integration, because in many cases, workflows, we do see the benefit of implementing different functions on a chip, not just dispense, but mixing, pumping, filtering, heating, and thermal cycling. A lot of those we have implemented on our microfluidic architecture. So where we would benefit from that integration, we have reduced it to practice, and we created modules which could lyse cells. They could perform sample prep. They could do single cell sense and isolation, rare cell isolation, and ultimately bring what we love about doing this integration is that ultimately, when we actually isolate something that's really precious, we can actually get it out of the system really easily, again, by performing a dispense function. The third category of technology convergence we've been pursuing is leveraging many of our microfluidics assets and chemistry and porous media assets and knowledge from print, and kind of deploying it into life sciences by trying to actually manufacture very low cost tests. And that would be, for example, lateral flow assays, which is, I think everyone knows now what that is with the coronavirus pandemic. Lateral flow assays are very low cost, but they are typically not very sensitive, not always reproducible. And the pipeline for development for them is fairly long. And what we wanted to do is to see if we could eliminate some of the pain points associated with manufacturing, which requires a large numbers of analog instruments and optimization of all of those. Instead, what we asked, can we actually just have a dispense system, a microfluidic system that can actually print a large number of iterations and different designs with a single setting with that domain expertise that runs deep. So how do we democratize and accelerate introduction of lateral flow assays for a lot of different applications by creating the tools that can really quickly start going through different iterations of designs? And then bringing other elements of our knowledge on how do we now, with the precision tools that we have at microfluidics, create very advanced functions on the lateral flow assay, which overcome the sensitivity and reproducibility limitations of this device. And here I'm just showing one example where we're doing not just 2D, but 3D flow focusing of samples to very precisely printed locations for the reactions to occur. So these are the examples that I just wanted to highlight. We have many more, and I'm happy to jump into more conversations one-on-ones. But the key takeaways is we feel we excel in manipulating and delivering very small volumes of fluids. We have a large diversity of compatibility with fluids, and we can leverage our economy of scale that comes from the print business to really quickly make low cost and disposable consumables. High spatial resolution patterning capability, and very flexible mode of operation. We could do integrated, outside, or a hybrid system. And just one final comment is that we are not a life sciences expert, and we're always seeking partnerships in both clinical and pharma and research industry. So that's all I have for today. And thank you for your time, and I'm open to questions.
Yep. Thank you, Anita, for your talk. Great to see all the ongoing stuff at the HP. And let me see if there are questions from the auditorium. Let's see, is there questions? Let's see, one moment. Yeah, there's a question from Jay Morale. Hi, Dr. Anita, will you be offering your microfluidic dispense platform as part of a design service or foundry-like services? Would you provide links to the design rules and PDK?
So I can't speak for what business model we would pursue in the future, but today I could say that we don't yet have a foundry model. So our operating mode would be to partner or collaborate, and then it would be more of a one-to-one interaction, and we would work through that type of engagement. But the foundry model has come up several times in our internal conversations.
Yeah. It's one question from my side, Anita. It's like when you look at the sample sizes, I think it says that you can go down to 2 microliters. But then on the other hand, the common challenges in diagnostics are specificity and sensitivity. That means even if you can go down to 2 microliters, how relevant would that be in the clinical practice?
Yeah, so we can go down to 2 picoliters. Just wanted to make sure that to make that correction. 2 microliters is actually a fairly good and large quantity. The 2 picoliter, I bring up that example is because in some application you want that type of resolution, but you might not operate with a 2 picoliter volume of solution. So if you're inputting, let's take as an example the PCR and ELISA workflows, the robotic automation system typically go down to 1 microliter, right? So that is actually fairly, you're bringing up microliter.
Yeah.
But that itself has been a limitation to many of these workflows, because you want to actually end up with a 1 to 2 microliter final reaction volume. So if you have to put in 1 to 2 microliter of sample, then you're essentially now scaling up your volume to get the ratios correctly. And oftentimes our partners say that when they're processing animal specimens or cell samples, if they have to have that high of a volume for an input, they have to start pooling their samples together. So having the ability to to discretize your volume, and having that resolution gives you the ability to scale down your reaction volumes, but without compromising on sensitivity. You need to know how much material you'll need to have relevant information at the end.
Very good. Yeah, this makes sense. That's a good thought. Any other questions for my co-chair?
I have a question, if you don't mind, Anita.
Yeah.
So, the idea of manufacturing on demand drugs has been around for a while, and it's been. What I'm kind of curious now, when you think about the technological elements needed to do something like that, and then of course there's the quality elements, there's the mostly quality regulatory elements that go into that.
I was going to add regulatory elements, yes.
Right.
Yeah.
So I mean, those are barriers that they're non-technological. But is there something that you can see that when you look from HP looks in the crystal ball to say, hey, you know what? Based on what we're anticipating in terms of changes that are going to happen in society, healthcare practices, and such, this could become a reality, and that's why you're positioning the way you are right now to intercept that at some point in the future? Or is it just something that's a good idea that still has to be? I mean, there's also a COGS element, cost of goods element to this as well. So I was kind of curious, is that realistic, or is it something that's kind of nice, but, still kind of aspirational?
Those are really excellent questions, and it warrants probably a whole hour conversation on this.
Yeah, no, we can't afford that, but.
No, exactly. I would just want to say that it's definitely in our forefront of thinking, because the regulatory aspect is of course very tricky. Taking a step back, for example, manufacturing drugs near patient completely from scratch does not make sense necessarily, unless that aspect of it, some customization, some personalization adds significant value and you can prove it. So in the case of the customized, like the tablet that I showed on the screen, I would say there are, for example, there's so many opportunities that have been highlighted in the past on this. But one simple example is, if you have a grandfather who is having a hard time taking their medication, right? They have 10 different ones, and every day they have a different numbers of pills to take. If you can provide a customized form factor, which includes all the necessary composition and the right interactions between those drugs in a tablet form, that particular part of the manufacturing maybe makes sense to deliver closer to the patient, but not the entire manufacturing, not making the compounds specifically. Or the core of the fundamental active ingredients that will go into that tablet, that they would not be made at the local site. But the same thing for personalized gene therapeutics. It's not the future anymore. We are in a space where FDA has already approved several personalized gene therapeutic solutions and products. So they are working through these regulatory challenges. What needs to happen in the manufacturing floor, the quality control system, the quality management systems to ensure safety, patient safety on a highly customized drug. So I think these are very important questions, but I would say within the limited time we have, it's a very worthy conversation and a very worthy battle to go through, because they clearly add value. That's why we're all talking about it.
Exactly right. Thank you.
Okay, all right, very good. I think this was a great session today. Thanks, Anita, again for joining.
Thank you for the invitation.
Sure thing. And then I actually want to conclude our session today. So we started a few hours ago, our precision medicine data science session. Quite nice overview, I think, starting with talks about predictive analytics, how to improve critical care, how also to turn cancer treatment into more immersive patient experience, as chemotherapy is a pretty complex program on its own. And it got all the great talk, great insights, I think, about the new advancements in cancer treatment, how to find new drugs. And I think we had also really nice zoom into specific examples of lung cancer to look at the mechanistic details. And then also, I think that especially like the last two talks had been great about showing the manufacturability, scalability aspect, right? I think showing how to scale biofoundries, how to make it scalable and affordable, and then also concluding with Anita's talk on the technology tools, how to basically make the immersive experience of chemotherapy or theranostics, for example, really possible with having really these type of highly scalable miniaturized testing devices. And that's all, I think, a great, I would say, manifestation of the technology convergence we are experiencing and witnessing, especially these years with the COVID-19. And I want to thank all speakers, all attendees. I want to thank also Dr. Akai and Dr. Colin Brennan for organizing this session together. Thank you very much.
Thank you so much. This was highly inspiring talks. We appreciate the speakers and look forward to seeing you all either in Italy, Capri Island, or in Israel for the next one. I'm praying for the face-to-face one because that's what our intention. And also, I would like to appreciate all the efforts by Ali and Colin. And we are going to build on the data science and engineering, healthcare or medicine biology. And I will be in touch with you. And this is also the part of the my presidential efforts to build really ecosystem. And we should get more clinician and practitioner industry leaders part of our engineering society. And we learn a lot from the clinician, and also we offer a lot with engineering innovation. I think it's, and this is, we will continue the public forum to increase the awareness of the healthcare innovation and the grand healthcare challenges. And also, hopefully we will get more students to participate. They help them to build their careers and also involve and interact with the industry leaders. Thank you so much. And look forward to see you tomorrow. And we will start at the same time. Tomorrow's symposium will focus on digital healthcare. And also on Saturday, we will focus on brain and neural systems. Thank you so much. Thank you. Please stay safe.
Thanks, everyone.
Thank you very much. Bye-bye.
Bye.
Take care.
Bye.